with learned professors and personals from different backgrounds to come together and to think what can be done to make science reach the common man. I feel very honored to have our learned speakers who have accepted our invitation for sparing their valuable time with us in this 2 days webinar. So we have Dr. Prakash Bisen from Gwalior, a learned person, scientific advisor, professor emeritus, and adjunct professor. Then we have Dr. Pramod Rath, professor, JNU Delhi, Dr. Dinesh Mishra, ophthalmologist, Raipur, Sadhna Sharma, he is also a social worker, Korba, Chhattisgarh. Dr. Varnika Sharma, he is doing his work in military psychology, a very important aspect. And Secretary Fans, Mr. Shashank Gupta, Dr. Anil Kumar, he is from Bihar, and Louis Mina Barriot, professor, he is professor of neuroscience from Brazil. So I thank you all from depth of my heart for accepting our invitation and welcome you in this 2 days webinar. I hope that this webinar will be thought-provoking and interactive, and we would come out with new ideas to approach society with scientific temperament. Please join with me giving a warm welcome to our speaker. Thank you very much.
Thank you so much, ma'am, for enlightening us with the overview of this webinar. Now, I am gladly requesting our respected principal, sir, Dr. P.C. Chaubey, for the inaugural address.
हेलो
Yes, sir.
मेरी आवाज आ रही है?
जी सर.
Yes, sir. जी sir.
Yes, sir.
Yeah, I welcome keynote speaker Dr. Prakash Bisen sir, Professor Jivaji University Gwalior, Dr. Pramod Rath, JNU Delhi, डॉक्टर दिनेश मिश्रा सर, मैं समझता हूँ अभी सर शायद जुड़ने वाले होंगे, डॉक्टर अनिल कुमार, प्रोफेसर जेपी यूनिवर्सिटी बिहार, साधना शर्मा, डायरेक्टर एमएलसी कॉलेज कोरबा, मिस्टर सुशांत शर्मा सर, फॉर्मर डायरेक्टर छत्तीसगढ़ साहित्य अकादमी, छत्तीसगढ़ साहित्य आई थिंक हिंदी ग्रंथ अकादमी, and Dr. Varnika Sharma, and all participants and all my colleagues. The main objective of the current SSR guideline is to harness the latent potential of the scientific community to strengthen science and society linkages by bridging. Science and society share a mutualistic relationship, and SSR will comprise the scientific impact on society and social implication for science. I sure that you all will feel enriched with knowledge after completion of this webinar. I wish to success this webinar. Thanking you to all.
Thank you, sir, for your insight and kind words of encouragement in all our endeavors. Your support and guidance is very precious for us. I now request our IQAC coordinator and convener of this webinar, Dr. Renu Maheshwari ma'am, to speak about the scope and objectives of this webinar.
Thank you, Dipali. Can I audible?
Yes, ma'am, you are audible, ma'am.
Okay, thank you. Good morning to all. Sorry, good afternoon to all, esteemed guests, participants, and my dear friends. I, Dr. Renu Maheshwari, convener of this webinar, welcome to all of you for this national webinar on scientific social responsibility. Science and technology have been an essential component of Indian civilization and culture over the many millennia. आजादी के बाद से भारत ने विज्ञान और प्रौद्योगिकी में उल्लेखनीय प्रगति की है. According to National Science Foundation database, India is placed in the third among countries in scientific publication. Despite making a good progress in science, technology, and information, the transfer of scientific knowledge and its benefit to society remains an area of concern. Now question, why is it? because of pandemic has served as a reminder of the importance of science, technology, and innovation policy in fostering resilience and facilitating shock adaptation. Prior, science and technology policy placed a strong emphasis on applying science to improve public welfare. लेकिन अगर हम दूसरी ओर देखते हैं, जहां हमारा देश भारत अपनी ऊर्जावान युवा आबादी के साथ महत्वाकांक्षाओं और बढ़ती आकांक्षाओं का देश है, जिसके लिए समाज के साथ विज्ञान एवं टेक्नोलॉजी के संस्थागत और व्यक्तिगत एकीकरण पर नए सिरे से ध्यान देने की आवश्यकता है। यह सुनिश्चित करने के लिए वैज्ञानिक ज्ञान और उसका लाभ समाज तक पहुंच सके, विज्ञान और समाज के एकीकरण को सुविधाजनक बनाने और स्टेक होल्डर्स के बीच तालमेल को बढ़ावा देने के लिए एक रूपरेखा विकसित की जानी चाहिए। 2017 में आयोजित 104वीं 104th इंडियन साइंस कांग्रेस ने समाज के लाभ के लिए विज्ञान को शामिल करने के लिए साइंटिफिक सोशल रिस्पॉन्सिबिलिटी को स्थापित करने की आवश्यकता पर जोर दिया, क्योंकि विज्ञान और समाज के बीच संबंध दो तरफा जुड़ाव है। साइंटिफिक सोशल रिस्पॉन्सिबिलिटी न केवल समाज पर वैज्ञानिक प्रभाव के बारे में है, बल्कि विज्ञान पर सामाजिक प्रभाव के बारे में भी है। Therefore, scientific social responsibility would bring efficiency and an attitudinal change in the mindset and the work style of the scientific community. Also would strengthen the knowledge ecosystem. Thereby, scientific social responsibility enhancing the social outreach of our scientific community. Teachers and scientists across all scientific and technology domains have an ethical duty, that is social responsibility, to freely share their expertise and resources with society's stakeholders as possible, acting with a spirit of service and thoughtful reciprocity. On this objective, our college has planned to organize a webinar on scientific social responsibility held on 19th and 20th January 2024. In this conference, there are 4 technical sessions. In the first session, keynote talk on food and human health is given by Dr. Prakash Desai from Gwalior. There are 3 speakers in technical session 2. First speaker, our special guest, Dr. Pramod Rath from JNU, Delhi, will deliver a lecture on inspiring students in science education research to make a better society. Second speaker, Dr. Dinesh Mishra, President of Anshraddha Nirmulan Samiti, Raipur, Chhattisgarh, will deliver a lecture on scientific temperament and social superstitions. Third speaker of this session, Dr. Sunita Patra from Dr. NPG College of Science, Raipur, will talk about the sickle cell awareness and scientific approach. There will be 2 technical sessions on second day of webinar, that is 21-24. In technical session 3, there will be 3 invited talks on different aspects of scientific social responsibility. First talk on how science impacts on our daily activities will be delivered by Sadhna Sharma, Academic Director, MLC College, Korba, Chhattisgarh. Dr. Varnika Sharma will talk about social direction of science and youth as a second speaker of this session. The third speaker, Mr. Shashank Sharma, a media person, will talk social and cultural studies in science. The fourth session of the webinar is of valedictory function, in which 2 guests of honor, Dr. Anil Kumar from Bihar, will talk on opportunity in diversity, and Dr. Luis Menna Barreto from Brazil will talk on social implication of chronobiology. The deliberation of this webinar will be helpful in addressing not only the direction of research to gain immediate application, but also will identify gaps in our knowledge in the domain of scientific social responsibility. Once again, I welcome all of you. Thank you.
Thank you so much, ma'am, for sharing your knowledge and information with us. With this, we may head towards our keynote lecture. We have Dr. Prakash Bisain, Honorary Emeritus Professor, School of Studies in Biotechnology, Jivaji University, Gwalior. Welcome, sir. It's a privilege to have you with us. I request Dr. Savita Singh, ma'am, to formally introduce our speaker. Dr. Bisain, sir.
Good morning, Pranam, and Jai Johar to all present on this virtual platform. It is my proud privilege to be introducing the keynote speaker today. And I'm very happy to say that he is an alumni of our college and he has been with us from 1967 to 69 and it is graduation from here. Welcome you, sir.
Thank you so much.
Sir. So, sir has got a very, very impressive profile, and I'm proud to read it. Was born in Jhansi and received his till 65. He was in Jhansi, received his PhD in 1972 and DSc in 1981 from Jabalpur University. He has guided 70 doctoral theses and has 350 research publications to his credit in indexed international journals of repute. 10 patents have been published by him in India, US, Europe, and Japan. 15 international books have been published by him on contemporary issues in biological sciences. On the administrative front, Sir has held many important responsibilities. He has been the director of the Institute of Microbiology and Biotechnology, Barkatullah University, the Vice Chancellor of Jivaji University, Gwalior. He's been the director of Madhav Institute of Technology and Science, Gwalior, and director and dean, J.C. Bose Institute of Life Sciences, Bundelkhand University, Jhansi. He's been the principal director and advisor at Jaipur National University, Jaipur. He was a fellow of the UNESCO, UNDP, ICRO, and Hungarian Academy of Sciences at the Institute of Plant Physiology, Biological Research Center, CZ, Hungary. He has been a part of the US National Science Foundation Fellow in the Department of Biological Sciences at the University of Illinois at Chicago and in the Department of Bacteriology at the University of California, Davis. He's been a UNESCO WHO Fellow at the Institute of Microbiology, Czechoslovakia, European Commission's Marie Curie Fellow at the Institute of Environment and Biological Sciences, University of Lancaster, United Kingdom, and DAAD Germany Academic Exchange Fellowship Fellow at the University of Bonn, Germany. He has received many awards. He was awarded the first Indira Gandhi Fellowship for his outstanding work on environment conservation and management. National Overseas Associateship of the Department of Biotechnology, Ministry of Science and Technology, Government of India. He's been a UGC Career Award, State Level Kailash Nath Kudzu Award of Government of Madhya Pradesh for contributions in life sciences. He was awarded the Lifetime Professorship, the highest honor of Bundelkhand University, Jhansi. He has also worked as Emeritus Scientist, CSIR, at Defense Research Development Establishment, Defense Research Development Organization, Ministry of Defense, Government of India, Gwalior, and is currently pursuing his research in the field of immune and nutritional therapy in medical biotechnology as an advisor in the Institute of Medical Sciences, Jaipur National University, Jaipur. And with the research project, he's working on a research project on tuberculosis from Grand Challenges Canada, Melinda Gates Foundation, Government of Canada, with a grant of Canadian Professor Bisain is the elected fellow of the National Academy of Sciences, India, for his enormous contributions to the field of biology. The diagnostic technology developed by Professor Bisain is unique in several respects, and he has received recognition worldwide. The technology has been patented in India, United States of America, Europe, and Japan. He has served as the external scientific advisor to a Barcelona-based Spanish multinational company, BioKit, involved in the manufacturing of diagnostic kits, and also serving as scientific advisor of an upcoming food industry, Troilite Foods, based at Gwalior, involved in probiotic research, food biotechnology, and which develops several innovative products for the food industry. Beijing doing the Institute of Translation Medical Group China and Nanyang Academy of Science Singapore have appointed him as senior research advisor. So we can see that he is got so many awards, and we can understand that he is a brilliant mind which is focused on scientific social responsibility, as he's working and linked with biotechnology, food diagnosis, and food products. And therefore, most appropriate and very, very gracious presence at this national seminar on scientific social responsibility. Sir, we are really honored to have you amongst us, and we are just waiting to listen to you. Thank you.
Thank you so much. सबसे पहले तो मैं साइंस कॉलेज के रिसर्च ग्रुप के कमेटी को मैं धन्यवाद देना चाहता हूं कि मुझे उन्होंने ये मौका दिया कि मैं am I audible? Can you hear me?
Yes,
Hello?
sir. Yes, sir. You're audible, sir.
मुझे मौका दिया कि मैं अपने पेरेंट इंस्टीट्यूशन में अपने थॉट्स, अपने विचार, शोध कार्य उन के साथ शेयर कर सकूं। 1967 में मैंने जब बीएससी की थी तो एक जूलॉजिस्ट प्रोफेसर आर.एन. सिंह हमारे प्रिंसिपल हुआ करते थे, और जब मैंने 1967 to 69 गवर्नमेंट साइंस कॉलेज में एमएससी में एडमिशन लिया था, तो प्रोफेसर एस.एम. अग्रवाल happened to be the head of the Department of Zoology, and Professor R.C. Yamunotri, he happened to be the head of the Department of Botany. तो साइंस कॉलेज के साथ मेरी बहुत सारी यादें जुड़ी हैं। अब ये करीब समझ लीजिए 60 साल का जर्नी है। 60 साल में इधर उधर रहा, और उसके बाद आज मुझे साइंस कॉलेज में बोलने का मौका मिला, और मैं जो कुछ भी हूँ, मेरी प्राइमरी ट्रेनिंग तो वहीं से हुई है। 1969 तक जो मैंने शिक्षा ली थी, उसी से मैं आगे बढ़ा, उसी से मैं आगे ये काम किया। तो फर्स्ट ऑफ ऑल, लेट मी थैंक एवरीवन, द प्रिंसिपल ऑफ द कॉलेज प्रोफेसर चौबे, द फैकल्टी, द मेंबर ऑफ द रिसर्च टीम, फॉर इनवाइटिंग मी टू शेयर माय थॉट्स ऑन food and society. When I talk about the food and society, there are three things which are very, very essential. One is the carbon, the another one nitrogen, and the third one is phosphorus. If there is no carbon, then there would be no carbohydrate. If there is no nitrogen, then there would be no functional or structural protein. And if there is no phosphate, then there would be no energy. So all these three things are very, very essential to survive, to sustain. And they are interlinked with each other. Now the carbon, where from do we get carbon? We get carbon from the atmosphere. And on the earth, other than plant, no other organism on the earth has got the capacity to fix carbon dioxide. Whatever carbon dioxide we emit during our actions that goes to the environment, and that carbon dioxide is fixed by the plant and converted into different forms of carbohydrate. It may be monosaccharide, disaccharide, polysaccharide, and starch. And that is what most of the animals they use for their food. But even this carbon dioxide, which is being fixed by the plant, because of the carbon dioxide fixation, will not act unless there are enzymes. Because the whole process that is operated through the enzymes, and enzymes are synthesized because of the nitrogen. The nitrogen gas.
Sorry, sir, for the interruption. Are you sharing some slide?
Yes.
Because we are not not able to see any slides here.
Okay, I will. Can you now?
स्क्रीन शेयर करना होगा, बता दो.
Sir, there is an option share screen and then you can go for it.
Can you see now?
Not yet. Yeah, yes, it is visible. Yes.
Okay.
Yes.
So,
Yeah, okay.
So basically the carbon dioxide is fixed by plant and the nitrogen is fixed by microorganism, the bacteria. Bacteria, they fix the atmospheric nitrogen and convert it into different form of amino acids and proteins. And the proteins, some of the proteins, they are the functional proteins, which is also known as enzymes. And some of the proteins that is known as the structural proteins like our skin. So both structural and functional proteins are synthesized by bacteria, and these bacteria, they are known as the nitrogen fixers. So the bacteria is a very essential component in our system. They fix the atmospheric nitrogen into enzymes, structural and functional protein, and the carbohydrate is the carbon dioxide is fixed by different type of enzymes to convert it into different polysaccharide, monosaccharide, and disaccharide. Whenever I start my lecture, there is a quote from Hubbard: "Man is the most insane species. He worships an invisible God and destroys the visible nature, unaware that this nature he is destroying is the God he is worshipping." So this is the very famous quote from Hubbard, and that is what happened at the time of COVID in 2020, the year of the rat. We were all in hiding. We only go out to get food. We store food to eat later. When people come close to us, we run away. This is what the reality was when there was a pandemic of the COVID-19. Now, the food is normally used for our sustenance, for nourishment, and for enjoyment. Looking for non-nutrient food component for added benefits from their food, such as health promotion, disease prevention, and performance improvement. They also prevent disease to ensure good health and longevity. Now, whatever the food we eat food to have sufficient energy. We eat food to have the enzymes activated, enzymes synthesized, and the reaction is performed. Now, when we talk about the carbohydrate and nitrogen, they provide basic nutrition and prevent disease to ensure good health and longevity. This is known as the functional food. The functional food is that which regulates the different functions in the body. The concept is not new to people in some parts of Asia. We believe that some food is beneficial to health, having therapeutic ability based on centuries of tradition, to which we say Ayurveda. Largely lacking scientific basis, therefore, there is a problem in accepting our views. Now, whatever food we eat, when we say the functional food, there is a reaction which is known as LADME, L-A-D-M-E. The LADME means the food what we eat, that has to be liberated, that has to be absorbed, is equally distributed, metabolized, and then the last phase is the elimination phase. So these are the very important aspect of functional food. And that is how the food is available to the system. So the functionality and bioavailability of bioactive compounds are the key steps in ensuring bioefficacy of bioactive food compounds taken orally. When we talk of functional food, that include different minerals, vitamins, fatty acid, dietary fibers, phytochemicals, antioxidants, and probiotics. So these are the important things which has to be there in the functional food. And that food with added biologically active substance, such as that they have lived beneficial cultures. Now, because of the food, because of our habit, because of our style of living style, if we do not follow the system, if we do not follow the rules of the nature, then there are viral and bacterial diseases. They start occurring in our body: cancer, coronary heart diseases, strokes, diabetes, atherosclerosis, and liver disease. They are all known as lifestyle diseases. If we change our lifestyle, then one gets any of these diseases which are known as the lifestyle diseases, or maybe the combination of more than one type of disease. There is a saying by Hippocrates, 400 years before Christ: death sits in the bowels. A bad digestion is the root of all evil. Intestine is a paradise of disease. So the root cause of disease is the intestine. And every type of disease that starts from the intestine. So if you control the intestine, if you take appropriate food, then probably you can save your body. Now, normally, whenever we talk about, we are scared of bacteria, we are scared of viruses. We say, oh, bacteria, they are very harmful, they will kill the body. But do you know that even less than 1% bacteria are pathogenic? 99.9% bacteria, they are all beneficial bacteria. In a body, we have trillions of living bacteria in the human intestine. And there are 100 trillion microbial cells in human body: mouth, intestine, vagina, and skin. 10x the number of human cells in our body. 10 times more bacteria are there than the human cells. And more than 500 different bacterial species they inhabit in the intestine. And if you talk about the weight, a normal human body has got 3.5 pound body weight consists of bacteria. So whatever your body weight is, you just eliminate 2.5 kilo, that is actually the real weight, which is more than the weight of brain and heart. Brain and heart, they together, they constitute 3.5 pound, and the bacteria constitute about 3.5 pound in the weight in human system. Now, the gut colonization that begins immediately after birth. The baby, when is born, is completely, maybe very less number of bacteria are there, but slowly it takes 18 to 20 months or 24 months to colonize the intestine of a baby. Colonization pattern is affected by mode and type of birth delivery. and initial diet, geographical location. Initial bacterial colonization starts from germ-free intrauterine environment and is populated through maternal, vaginal, and fetal flora and feeding breast milk versus formula. So that all constitute the bacterial population in a gut. Now, when we talk about the gut bacteria, I think you all know as a student of biology that human system has got two brain system, two nervous system. One that is known as enteric nervous system, and the second one is the central nervous system. Central nervous system is connected with your enteric nervous system through gut-brain axis. And in the human body, there are human virome. Number of viruses are located in the body, in intestine, in your blood, and throughout body, there are trillions and trillions of viruses are present in the body. And these viruses, they infect human cells, they infect microbes, they infect bacteria, they infect fungi if fungal cells are there. Those viruses which affect the bacteria, they are known as bacteriophage. They kill the bacteria, they lyse the cells of the bacteria, and when the body becomes weak, then some of the viruses, they infect the human body and create number of different viral diseases. So very few viruses, they affect the body. If the immunity is weak, if the immunity is compromised, they cause disease. And certain viruses, they integrate with the human genome as proviruses. So some of the viruses, they become part of the human cells, and then they express themselves as a provirus. They infect and cause disease and are affected by pre-existing immunity. So in fact, if there are no viruses, then probably in the human body will be subjected to attack by various microorganisms. They provide immunity to the body. Unlike roughly 40 trillion bacteria in a typical human microbiome, exact figure of viruses in a human being is not known till date. The human virome is not stable and may change over time. New viruses are discovered constantly, and the bacteria and viruses in the human gut can be changed by change in diet. Blood virome will be different from the gut or skin. Now, when I talk enteric, second nervous system in the human body, newly discovered protein explaining power of gut bacteria. Now, the gut bacteria, they pass signal to the central nervous system through the nerve, which is connecting brain with the alimentary canal, with the gut. And in the past few years alone, studies have linked gut flora to Parkinson's, autism, schizophrenia, and many more health conditions. Now, the people have come to a conclusion. There is a recent paper in 2018 that they have got some clue that the Parkinson disease can be controlled by treating or by treating with the probiotic bacteria, by enhancing the bacterial population, probiotic bacterial population in the human gut. And this axis is known as gut-brain axis connection, which explain how overeating leads to obesity. Bacteria injecting help the immune system fight cancer. Your gut is directly connected to your brain by a newly discovered neuron circuit. And this neuron circuit is known as enteric nervous system. Now, the enteric nervous system is too thin layer of more than 100 million nerve cells lining gastrointestinal tract from esophagus to rectum. Its main role is controlling digestion, from swallowing to the release of enzymes that break down food to the control of blood flow that helps with nutrient absorption to elimination. The enteric nervous system doesn't seem capable of thought as we know it, but it communicates back and forth with our big brain with profound results. Signals from digestive system affect metabolism, raising or reducing risk for health conditions like type 2 diabetes. This involves interaction between nerve signals, gut hormones, and microbiota, the bacteria that live in the digestive tract. Now I come to the gut colonization. Now these are the different type of bacteria, and this is the vaginal delivery, and this is the cesarean delivery. Now this is the different population of bacteria in the colonization when it takes place in the intestine, and which is different from the cesarean delivery system. So the bacterial population which develops in a baby after birth is different with the normal delivery than when you compare with the cesarean delivery. Now, the human gut contains trillions of microorganisms. In fact, it's home to 10 times more bacteria than human cells. These residents play a remarkably important role in our body, from boosting our immune system to even affecting our behavior and personality. Tens of thousands of proteins created by the bacteria that dwell in the mouth, gut, skin, and vagina of humans. The new insight could be used to harness the power of human microbiome, paving way for new treatment and drugs. It's critically important to understand the interface between human cells and the microbiome. The human microbiome and their wider effect on health is still elude full understanding, through a mounting heap of evidence is showing that the composition of bacteria in the intestinal tract could have far-reaching effect on your health. Now, the role of gut microbiome in the human diseases, very promising results have come with inflammatory bowel diseases, with cardiovascular system, with obesity, with autoimmune diseases, with cancer, liver disease, many other, and Alzheimer and other brain diseases. So this is all the role of probiotic bacteria. When I talk about the functional food, now whatever the food you eat, that is important for the multiplication of the multiplication of the probiotic bacteria. The probiotic bacteria, they are also known as lactic acid bacteria. The lactic acid bacteria are those which secrete lactic acid, and the food or the substrate for their multiplication is lactose. And if you are eating carbohydrate, then, or if you are eating more of protein than the carbohydrate, then the colonization of probiotic bacteria in your gut would be different than the one which is developing because of eating carbohydrate. So both different type of bacteria, the colonization will be there. Now, the second thing is, well, in the initial lecture, I was talking about the functional food. Now, what is happening? We eat fruits. The fruits is known for the antioxidants, the color compounds. The color compounds are actually the antioxidants, and different type of antioxidants are synthesized in plants. There is a pathway which is known as cyclic acid pathway. The cyclic acid pathway is known for the production of different type of phenols, different type of terpenes, different type of flavonoids. So these are all synthesized in plant because of the cyclic acid pathway. And cyclic acid pathway is only in plant. Nowhere else you will find the cyclic acid pathway. and the cyclic acid pathway, they use different type of aromatic amino acid to convert it into different type of phenolic compounds like catechoric acid, caffeic acid, chlorogenic acid, lutein, hirsutein, anthocyanin, leucoanthocyanin. All these are the antioxidant compounds, and they are synthesized in plants, largely in flowers and in fruits, sometime in different portion of the plants. Now, when we talk about the fruits, now apple has got anthocyanin and cyanin. Apple is grown in temperate climate. Apple, whatever we eat, we get it from Kashmir, we get it from Himachal Pradesh, from colder area. We are living in the tropical area, and in the tropical area, we get guava, we get a different type of fruits than when you compare with the temperate fruit. The nature has developed us in a tropical climate. As per the requirement of the tropical climate, our body is adjusted with the tropical climate. So we will be more friendly with the fruits which is nearby us, which is growing. So always the people should be more dependent on regional, seasonal, annual fruits, whatever is grown in the local climate, rather than importing it from outside. Now, if we take the fruits from Kashmir, from Himachal Pradesh, whatever is grown over there, now those fruits for us is of no use, because our body is acclimatized with the tropical climate and can be very well adjusted with the fruits, with the food, whatever is being grown in Chhattisgarh, than the one which is growing in the temperate climate, or like in Himachal Pradesh, or in Kashmir, or in New Zealand, or in America. But since our economic condition has improved, now everybody wants to have the apple from New Zealand because we can pay it, but that is of no use. Now it is up to you to decide whether you want to use food as a medicine or medicine as a food. So the moment you will decide that you will use your food or fruit as a medicine, that will be more valuable than the one which if you are importing it from the outside. Now another thing is, we take oil. When we take oil like us, we are dependent on mustard oil, because here the mustard is largely grown, and mustard has got more amount of sulfur, and here the people, they can accommodate, or they will be adjusted more with the mustard oil than the coconut oil. Coconut oil is grown in South India, so there the edible oil is coconut oil. And maybe in Chhattisgarh you may have the peanut oil. Peanut oil is very famous. Groundnut oil is very famous for the cooking this. Now, for different type of diseases, what is happening in our system is that when we use oil, we normally use refined oil. What do we do with the refined oil? We just take out all glycerides, because of the glycerides in the oil, it becomes more viscous. When it becomes more viscous, then you see the advertisement on the TV that this oil is just like water without any viscosity. And you don't realize that this viscous portion in the oil, which is known as the glyceride, that is heart protectant, that protects your heart. And those multinational companies or the corporate houses, they just take out the glyceride and sell it as an ancillary product for pharmaceutical industry or for cosmetic industries, for cream or different type of medicine. So always use the natural product as it is rather than the refined one. Refined product that enhances the different type of cardiovascular diseases. The second thing is, when I was talking to you about the gut microbiome, about the probiotic. Now the thing is, these days, most of the people, they can afford to buy the probiotic or anything from Amazon. They will go in Amazon and buy the most expensive probiotic mixture. Now, when the probiotic you buy, there would be mixture of 6 different bacteria, 5 different bacteria, 10 different bacteria of different combinations, maybe 10 billion, 20 billion cells. Now, the people will prefer to buy a bottle of 100 capsule of probiotic bacteria, which is of American origin. American origin bacteria are of no use to you. They have been developed in a temperate country, temperate environment, cold atmosphere. Those bacteria, the activities of those bacteria will be different than the bacteria which you have from the tropical places. The best would be if you isolate the bacteria, if you have the combination of bacteria of your region, that would be more useful. I am telling you of my own experience. I was invited in a meeting by DSIR, Department of Scientific and Industry Research, at Ministry of Science and Technology, Government of India, and they were discussing that we are importing culture worth billions of dollars from Canada, from Italy, from Scandinavian countries, and those bacteria, they were given to different dairy industries, which were responsible for making curd, making different type of milk product. That was my first experience. Then I said that in India we have the tropical climate, we have the temperate climate. When we have both the type of climate, can't we isolate these different probiotic bacteria from India? Then they said to me that we have a problem. If you have some idea, you submit a project and we will fund you. I was already a retired person, so I said that I will try to associate myself with any company, and if they agree, then we'll do it. Then I associated myself with this Triple Food, which was actually the food industry, the formulation industry. I asked them that if I start a R&D on isolating different probiotic bacteria, will you allow? Then they agreed and they appointed me as a senior scientific advisor in the company. And I was assigned the job to isolate the probiotic bacteria. Now, I am telling you my own experience in the process, in the journey. I started and I took 2 reference samples, and those samples were from Canada and Italy. The curding of milk usually used to be in 4 or 4.5 hours with those imported cultures. And I isolated bacteria from cow milk, from goat milk, from buffalo milk of different varieties, buffalo, different varieties of cow, different varieties of goat. And the curding used to take maybe 6 to 8 hours for curding of milk. And ultimately, at last, after a year of research work with no success, then one day I thought that why should I not try to isolate bacteria from camel milk, from Bikaner, from desert, Indian desert. And from there, when we took the milk sample and isolated bacteria, they were much better than the imported bacteria from Canada and Italy, and the curding used to take place within 3.5 hours because they were from the tropical area, and because of the tropical area, they were more effective than the one which we were importing. So, in the
Excuse me, sir.
population of whether it is a
Excuse me,
probiotic
sir.
bacteria, hello,
Sir, sorry to interrupt again. Sir,
yeah,
your slides are not moving, sir.
okay,
So please put it in slideshow mode, sir.
yeah, can you
That
see
will
now?
be okay. Yes, sir. It is now moving, sir. Okay.
Okay, now
Okay. Thank you, sir.
these probiotic bacteria, they have different functions, that is protective functions, the pathogen displacement, nutrient competition, receptor competition, production of antimicrobial factor, for example, bacteriocin and lactic acid. And these are known as the commensal bacteria. This is all I am telling you of probiotic bacteria, I think that in future, this is the system which is going to replace your chemotherapy. Although it may take some time, maybe a few years, but ultimately this is the system which is going to generate. And these probiotic bacteria, you have recently seen in the COVID-19 that our vaccine system has failed. Our vaccine system has failed. And these bacteria, they act as live vaccine. Because in your body, there are trillions and trillions of bacteria. My body will require some different type of immune functions will perform, and the another's body will different type of immune system will perform. So in my body, those bacteria which is good for my body to respond to immune system, that type of antibodies will be generated, which will be different from any other person. Or if I am sitting at Chhattisgarh, then my immune system will be different than if I am sitting at temperate places. So these are the different functions which are performed. Maybe some probiotic bacteria may change the barrier fortification, induction of IgA, apical tightening of tight junctions, immune system development, and then the metabolic functions. These are the three different functions are performed by probiotic bacteria in the intestine, in the gut, the protective function, structural function, and metabolic function. In metabolic function, the requirement of magnesium, calcium, and iron that is released, and that vitamin K, biotin, and folate that is also released. They get it, and then the short chain fatty acids that is also released. So these are the three different functions are performed in the gut by the probiotic bacteria: the protective function, structural function, and metabolic function. Now, what factors affect the intestinal microecosystem? As I said to you, the type of food which you eat, that ultimately colonizes different type of bacteria in your gut. And as I said to you in my first slide, mother's microbiology, that influence the microbiology and then the gut microbiology of baby, mode of delivery, and feeding practices. On mother's milk or on formula milk, there would be different type of gut microbiota in a baby. Later in life, antibiotic and other intake. If you take antibiotics, that will disturb your microbial flora in the gut. Infection,
Mute yourself.
processed low fiber, chronic diarrhea, stress, and chlorinated water that will affect the probiotic condition. So these are the different types of the hindrance for the microbial population to go to multiply. Why are probiotic important for human health? Probiotic is very important. And the antibiotics, alcohol consumption, and stress, they directly destroy the gut microflora. Therefore, whenever for any ailment you take antibiotics, your digestion is disturbed and your mood is off. So now here you see that the signal which has been passed from your enteric nervous system to central nervous system, that is affected. So that is because of the problem in digestion. Emergence of antibiotic resistant strains. Sometimes what happens, some strains, they become resistant to antibiotic. And then again, the problem in absorption of nutrients in the body, that starts. These are the different type of probiotic bacteria which are normally found in human gut. Why human gut? You will find them in the gut of any animal or maybe even in the birds too. Lactobacillus, Basidiophytobacterium, Streptococcus, Thermophilus, Saccharomyces, that is fungi, yeast, and Propionibacterium, Bacillus, Enterococcus, and Escherichia coli. These bulk single culture range, we have nearly 2,000 different type of probiotic bacteria which is being sold to different companies and different combination, and these are the bacteria which are normally used. Mechanism of action, bioconversion of, for example, sugar into fermented products, production of growth substrates like vitamin B and K. For other bacteria, direct antagonism by antimicrobial substance, hydrogen peroxide, organic acid, bacteriocin, and acidophilin. Metchnikoff, he for the first time conceptualized the idea of this probiotic at the start of 20th century, Russian Nobel Prize winner and father of modern immunology. He gave the concept of this. First commercial product of probiotic came in the market, that was Yakult. Probiotic, they can be found both in supplement form and component of food. Now here I am again coming to the food. Now, different food, they have got different type of antioxidants, and they are all color compounds. Now, we have destroyed our food. We have destroyed our food. When we go to the shop to buy any food, we try to see that the expiry date is extended. This may be a good food because here the expiry is for 6 months, or here the expiry is for 3 months. Now, imagine that the food which has got an extended period of expiry will not be good for you. Simply because the preservative, whatever is being used to save the food from bacterial contamination, from bacterial degradation, fungal degradation, from fungal degradation, they use some preservative to kill bacteria, to kill fungi. Of course, they say that this is within the permissible limit. Now, when this preservative is added to kill the microbe population in your food product, they accumulate in your body. When they accumulate in your body, they are the cause of cancer. Ultimately, all these are having compounds which are killing bacteria. But at young age, your body is strong, you can resist, body can adapt, But at old age, the body is unable to resist the dose of this heavy metals or different type of chemical compound preservatives, then the body surrenders at the age of 60 or maybe at the age of 65, 70, and then ultimately different type of ailment that starts in the body. So one should try to avoid using the packaged food with different type of packaging material or with different type of preservatives. So resistant to low pH, organic acid, bile, juice, saliva, human in origin, ability to immune response. So these are the properties of the bacteria. For any type of good bacteria, for any type of good probiotic, there should be good addition. There should be good addition with the alimentary canal, with the gut, colonization to human intestinal tract and influence on gut mucosal permeability, antagonistic against carcinogenic or pathogenic organisms. So these probiotic bacteria, though they should be, sometimes they develop resistance against carcinogenic agent, or sometimes they detoxify the gut from different type of toxic compound. Clinical proving health benefit, for example, gastrointestinal disorders, diarrhea, Clostridium difficile colitis, antibiotic-associated diarrhea, and acute gastrointestinal tract infection. How do they act? What is the mechanism of action? The bioconversion, for example, sugar into fermented product. All this starch, disaccharide, polysaccharides, they are converted into simple sugar, fermented into simple sugar, fermented into ultimate product like maybe acetic acid and different things, which is absorbable by the body. Production of growth substance like vitamin B and K for other bacteria. Direct antagonism by antimicrobial substance like hydrogen peroxide, organic acid, bacteriocin, and acidophilin. Now, these are three different functions of probiotics. As I said, that there are one way of vaccine production. You inject the bacteria or the viruses into an animal system, and then you take out the blood, purify the antibodies, and that antibody that act as a vaccine. So this is the normal procedure, and in a normal way, it takes nearly 24 to 30 months for developing the complete vaccine. This is for the first time with the COVID, in the history of vaccine development, that the last vaccine against COVID-19 was developed very quickly in 3 to 4 months, and stating that we have developed a technique through messenger RNA. So instead of following that, we have developed a technique with the help of messenger RNA. We have developed this vaccine, and which took maybe about 3 months or 4 months time. And you see the result that is not effective. Then after vaccination, you have to take one booster, you have to take then the second booster, and number of people, they got the infection of the COVID-19 even after the vaccination also. Now, in the case of probiotic, in the case of your food habit, if you take proper food which supports the probiotic growth in your gut, then what will happen? They will support the proper type of probiotic which will grow in your body. And when they will grow in your body, there will be number of different billions and billions of different bacteria. They will multiply, they will grow in your gut and contain some of the bacteria, and they will fight against your the body, and they will act as antigen. The bacteria, probiotic bacteria, they work as antigen, but we don't realize their importance. Now the probiotic bacteria are being given as replacing the chemo agent for the brain diseases like Alzheimer, like your dementia. And this paper, if somebody is interested, go and see in Science of 2016, 2018, the successful experiments they have reported that with the enteric nervous system, they are treating the ailment in the central nervous system with the probiotic combinations. So in normal intestine, they mucosa, say they regulate the bowel movement, and then they strengthen the immunity, and then the metabolic effects that the lactose intolerance, simplify the carbohydrate, simplify the complex protein into simple amino acid or simple peptide bonds, which are absorbable easily. Now, as I said in the early, that the disease rests on the intestine. If your digestion is not good, then ultimately that creates a lot of problem. And this is acute diarrhea and traveler's diarrhea, difficult lactose digestion, different type of colic infections, allergy, and growth parameters, undernourished children, cold, respiratory infection, vaginal infection, metabolic syndrome, skin inflammation, dental failure. So all these things happen, and they can very well be treated with probiotic. I mean, they will, the probiotic, what will do? There are two things. One is protection, the another one is defense. The probiotic will help to strengthen your defense mechanism by providing the appropriate antibodies to protect your body against various infections or against various ailments. Now, whenever we give probiotic, in probiotic, you have to give a supplement. A supplement, in supplement means stable, to stabilize that. And for stabilizing the bacterial cells, non-digestible food ingredients that beneficial affect the host by selectively stimulating the growth and activity of one species or a limited number of species of bacteria in the colon. And for that, we use sometimes oligosaccharides, inulin, fructose oligosaccharide, fiber, and fiber supplements. So they provide stability to the probiotic. Now, as I was discussing with you, the Parkinson disease, the neurons that make up your enteric nervous system, the brain in your gut, the brain in your gut are almost entirely replaced every 2 weeks. A study in mice suggests what's more an imbalance in the gut ability to repopulate itself with new neurons and clear out the dead ones could lead to Parkinson disease. So far in the cancer, we say apoptosis. You know about the apoptosis, that older cells, they die and the equal number of new cells are regenerated. So that way, the active cells balance that remains as it is. The moment the cells, the new cells, the formation of new cells are more than the dead cells, then what happens ultimately? The tumor is created because the new cells, they create tumor. There is an imbalance in the cell multiplication with the new cells and old cells. So if the new cells are more synthesized than the dead cells, then the tumor is created. And that process is known as, if it is remain as balanced, that the new cells are synthesized and equal number of cells are dead, then it is known as apoptosis. Now, with the Parkinson disease, even with the neurons, they have discovered that the new neurons are replaced, the old neurons are replaced by new neurons after every 15 days, after every 2 weeks. And that is how they make the complete system in operation in a normal way. So now with that brain gut axis, the treatment through the gut microbiota or through the probiotic bacteria, they were able to control the Parkinson disease. If anybody wants, we can read this paper in New Scientist. This article, this is a complete article and beautiful article. One must know about the role of enteric nervous system. Now one may ask, what is the difference between enteric nervous system and the central nervous system? Almost same. There is no difference except that with the brain, we have a thinking process. We have a thinking process, and the enteric nervous system, they do not have the thinking process. Otherwise, the system, the number of neurons may be more in enteric nervous system than the central nervous system. Now, in conclusion, I can say the probiotic bacteria, the food, the fruits, they all are very essential component for making a healthy human society. When I talk about the healthy human society, I talk the healthy by not only physical, by mental also, and that ultimately the food is a very important component. And instead of depending on exotic food, one should rely much on the local food, on the food which is grown in your own area. Like in Chhattisgarh, I like that the basa they prepare what is fermented food with the rice. That is good. I mean, the people in the village they used to take this basa, and that is good because it ferment the food. The rice is fermented, and some alcohol and number of bacteria are generated, and that ultimately when you take it, then all those bacteria which is of local origin, they go in your gut and they protect your gut and they perform their function to develop your immunity, to liberate the food, the bioactive compounds. Not all probiotics are equal. Some probiotics, not all probiotics. A probiotic may be more effective in my body, may not be effective in another's body. They will require B type of probiotic. It's like, but they are all living vaccine. They will create and they will develop the antigen in the body as per the body's requirement. They disconnect between scientific evidence and allowable claims. Still, the scientific knowledge is enhancing. People are working at different places, and NIH is actively involved in this. And particularly at this juncture, more people are working on obesity and on nervous system, the disorder in the brain. Claims should be substantiated with well-controlled clinical studies. They are already undergoing, and product should be characterized for content and stability. So this is all what I wanted to say, that one should rely more on the local food, more on the local sustenance and the local food, local probiotic bacteria, local lab. That is what is more important. And if you have a healthy society, and healthy society can be formed with healthy life, mentally and physically. So with these words, I close my lecture. And if there is any question, I will be very, very happy to reply to your questions. Thank you very much.
Thank you, sir. Participants, you can ask questions if you have.
So it was nice doing a webinar with you because we are celebrating, a science college is celebrating Diamond Jubilee year. And alumni like you who has actually emotionally connected to this institution, we are really honored to have as a keynote speaker. I really want to thank that.
I expected some questions from the audience on probiotic, but like you all know much about the probiotic.
I have one question, sir. So
Yes.
will this lead to like customized medications for individuals, sir? Like if we have different gut microbiota and like if the medications has to be customized on the basis of the individual?
Yeah, you mean the
The
specific
treatment plans I'm asking.
eh?
Treatment plans for diseases like.
Treatment now these days, what is happening? One more thing I wanted to emphasize that when doctors you go for any ailment, for any disease, sometimes the doctors, I mean, they are well trained, but they are not well educated. What do? They give some of the doctors may not be always everyone. They give antibiotics and probiotic together. So they will say that you take this antibiotic because of any ailment, and with this once or two capsule of probiotic. Now, as I explained in my lecture, that antibiotics will kill bacteria. So you are giving probiotic. Probiotic cannot be recommended with antibiotics, or antibiotics should not be recommended with probiotic. This is one thing. Second thing is the probiotic is available in the market in the form of capsule or in the form of tablet. Whenever you go, you will find it in a tablet with different strength, and the strength is calculated in terms of qualitatively. Qualitative means 5 bacteria, 6 bacteria, 7 bacteria, and quantitatively 10 billion or 50 million or number of cells there. Now, the moment you take one, and doctor will recommend you take one capsule with water in a day or twice daily, that bacteria or the probiotic will not work. Why? Because the probiotic is not a chemical. It is a life system. It has been lyophilized. It has been tabulated. Now you have to rehydrate it. You have to activate it. Now, in order to activate this probiotic bacteria, I perform my own experiment that you take capsule or you take the tablet, put it in warm water, lukewarm water of 35 degrees centigrade, which is used for preparing curd. Just incubate 5 to 10 minutes. So what will happen? This will perform two functions. One, the all probiotic bacteria, they are lab bacteria, lactic acid bacteria. Lactic acid bacteria means the substrate for these bacterial multiplication is lactose sugar. When you incubate with milk and with lukewarm milk, now that 30 or 35 degrees centigrade temperature, that will be required for bacterial multiplication, and the ideal temperature, plus it will have the lactose sugar, the substrate. So those cells, number of cells, they will in 5 minutes time or 10 minutes time, they will multiply, activate, and you take that milk. And if you take that milk, that will be more effective than the simple one capsule like antibiotic you are taking or probiotic. So this is my suggestion, that whenever you take, and there is a scientific basis, that you will provide them the substrate lactose, you will provide the optimal temperature required for their multiplication. That will be more helpful for the body to accept it to absorb.
Thank you so much, sir. So since childhood, we have listened to this, like you are what you eat. Like in our culture, we say Anna Purna Brahma hota hai. Now we have scientific reasons for that. Ki what you eat, you become that.
Yeah.
Yes. Thank you,
Yes.
sir. Now I invite Dr. Savita Singh ma'am to extend formal vote of thanks.
Thank you so much, sir, for your wonderful lecture on health, food and human health. It is something such a very, very relevant topic. And what we are seeing today, so many things that we do not even understand. We just follow things traditionally. But your lecture has been very, very incisive, very, very insightful. And we have definitely learned lots and lots. It has always been said that health is wealth and good food is like a good investment and all those things. But your lecture has given us lots and lots of knowledge, very deep, well researched knowledge. And your presentation was so very, very clear. We all know that health is very important, and how disease and health and therapy, all these that are related to food. You have given a wonderful lecture, sir. Thank you very much. Thank you.
Thank you very much. Once again, I thank the entire college team for inviting me, for interacting with you. My alma mater, where from I learned the science. And when I did my MSc from there were no microbiology, there were no biotechnology, there were no biochemistry. Only there were two subjects, zoology and botany. And those who goes for biology, they used to go for medical. Those who go for the mathematics, physics, and chemistry. But during the journey, academic journey, I learned about cancer and all these things. So that was actually, I was trained at Science College, Jabalpur, or at Arts and Science College, Durg. So I owe everything to the Science College. My principal was Rameshwar Shukla Anchal, and Rajeshwar Guru was these were the two principals when I was the student of the college. So a lot of memories. I thank every one of you, and particularly the principal and the entire team of Science College for inviting me for this talk. Thanks a lot.
Thank you, sir.
Thank you. Thank you, sir. Thank you, sir.
We can move on to our next session, ma'am.
Yes, we can move on to our next session.
Dr.
I think
Rath
Dr.
has
Rath
joined.
has joined.
Sir has joined, ma'am.
Yes, yes.
Yeah,
He
can
हाँ उन्होंने join कर लिया.
Okay, okay.
so can you hear me? Good afternoon.
Yes, sir. Yes, sir.
Okay, okay.
Good afternoon.
So I don't know if the picture, you have the video also.
Your video is not on right now, sir.
Oh, okay, okay, okay. So yeah, it should come. Yeah, I think, can you see the video now?
Yes, sir. Yes, sir.
Yeah,
Quite
yeah,
clear.
okay. Okay, good afternoon to all of you. So, yeah, you can
Good
go
afternoon,
ahead.
sir. Yes,
You
sir.
can-
Good afternoon, sir. Now
Yeah,
we are
you-
on the second technical session. We have Dr. Pramod Rath, professor from JNU Delhi with us. Welcome, sir. Sir will be speaking on inspiring students in science education and research to make a better society. I request Dr. Sunita Batra ma'am to introduce Dr. Rath, sir.
Thank you, Dipali. Sir, very good afternoon. I have the privilege to introduce you Dr. Pramod Seekers, who is a molecular biologist, and he has dedicated his career in studying gene expression through aging. Professor of JNU, he's basically a geologist, and he has studied gene expression during aging, and the model animal was a rat. Specialization was biochemistry. Research area of interest, that is genomic biology of repetitive DNA and non-coding RNA. Very interesting, repetitive DNA and non-coding RNA. Then cytokines, transcription factors, cell signaling, diseases, bone marrow stem cells, and regenerative medicines, and molecular biology of aging. I should really highlight some of the summary of his research work. Dr. Rath's team has isolated and characterized many mammalian complementary DNAs, cDNAs, with simple repeat sequences, and shown that genomic repetitive DNA and retrotransposable sequences are widely expressed into long non-coding RNAs in rat tissues. They have studied expression and structure function relationship of transcription factor, intron regulatory factors. They have also studied an interferon inducible antiviral endoribose. They have isolated mesenchymal stem cell from mouse bone marrow and has differentiated them into adipocytes, chondrocytes, osteocytes, neuron-like cells in order to study biology of stem cells. Very interesting. Their differentiation potential and its responsible application in regenerative medicines. At present, he is studying role of long non-coding RNAs in aging of brain and testes in rats and age-related diseases. He has published many journals, publications in reputed journals such as Molecular Biology of Aging, Bone Marrow Stem Cells and Regenerative Medicines, Genomic Biology of Repetitive DNA and Non-Coding RNAs, Cytokine Transcription Factors, Cell Signaling and Diseases. These are the very renowned journals in which the papers has been published. And there is a list of awards and honors. He has been Research Associate Fellow, then Vice President, Association of Gerontology of India. He has been expert members in DBT, Department of Biotechnology, JNU, UGC, Guru Nanak Dev University, Amritsar, in the National Agriculture Innovation Project Committee of ICAR, Karnal. He has been visiting scientist in MD Anderson Cancer Center, USA. Then in a NIH workshop, she got a scholarship in the project Advanced Genome Sequencing and Analysis, USA. And then he also a recipient of postdoctorate fellowship, Institute of Molecular Biology, Switzerland. He's also recipient of Tilak Award and A.B. Mishra Award. I think right from the MSc, is starting from the career, from postgraduate career, he is one of the brilliant student. And today, and it is the talk itself is very interesting and non-coding RNAs. So today we have Dr. Pramod Rath. He will be sharing his insights on inspiring students in science education and research to make a better society. I welcome you, sir. I'm Dr. Sunita Patra, organizing secretary of this webinar. Thank you very much. And you're very interesting. I was going through it. Thank you, sir.
So thank you very much, all of you, and especially Dr. Sunita, for your very kind words. So is it possible that we see the video of the audience also? Or
Video.
the audience, or all of
Yes.
them are joining online only?
They are joining online. Some of them are on YouTube.
Okay. So that's fine. So let me share my presentation. Then I think can you see the slide?
Yeah,
Yes,
yes,
sir.
sir, we can see.
Yeah. I think the top thing is probably covering it a little bit. So yeah. So can you see now? Yeah.
No, it is
No,
not
sir.
visible. It was no.
No, you can't see. Okay. a moment, let me fix it. Can you see now?
No, no.
Oh, okay. So let me, I think there's something wrong here.
It was shared earlier, sir.
Yes, yes, it is visible.
Yes, sir.
Okay, okay, okay. So I think, thank you very much for giving me a chance to talk to you. And is there some background noise coming? I think some audio is on probably. So as we know, this today's talk is basically inspiring students in science education and research to make a better society. So I don't know how many students are linked to this conference. So do you know how many students are there in this webinar?
Yeah, near about 85 participants there.
Oh, okay, okay. So they're all your students of your college.
Students and also faculties from this
Okay.
college and different colleges.
Okay, okay. So basically, why I choose this topic is this is very close to our profession and our heart. At JNU, the most important thing that we consider is how we train students along with teaching a subject or a course. So everybody comes here as a student to study, let's say, undergraduate program in some languages, and most of the students are actually postgraduate and PhD students in science. So with respect to life sciences, because I belong to the School of Life Sciences, we have basically master's program and PhD program. And one of the basic objectives of JNU is to catch students from most part of the country, including rural areas or deprived sections of the society, so that they get a chance to come to a good university and study their, whichever program they enroll. And through their study, they acquire knowledge and also get acquainted with their fellows, friends, and in an environment of the university, they sort of discover themselves for making their career to go ahead in future. So it's very important to get students from all parts of the country, not only good students from cities who have got already good education, but also students from remote places who are equally capable and talented, but probably do not have equal opportunity. So JNU gives this mandate to itself. Therefore, we consider this as an important aspect of education. And as a research university, it always gives focus on research along with education or teaching. And through education only, the students, they discover themselves, and especially with science education, and they decide what they will do in future. So if you see, can you see the next slide? Yeah?
No, sir.
No, sir.
Sir,
Next,
actually, it's not in slide mode.
you cannot see the slide?
Yes, we can see the slide, but the slides is not moving. It is still the
That is
first one.
slideshow mode.
But I am in the second slide now. So, okay, maybe I have to click this one.
Yes,
Yeah.
yes, we can see. Yes, we can see.
Oh, you
Yes,
can
sir.
see now. Okay.
Yes,
Yes,
yes.
yes.
So, okay. So, so the talk has different words, different aspects. One is inspiration, and the second is the youth, then student, then science, then education, then research, then society. So inspiration is something which we, can do so that the student or the young people actually like it, and they get motivated or inspired. So they start believing in what we say. So they develop a trust. So this is very important component, that we are actually on the same platform of thinking. And as we know that the colleges and universities are actually workshops of young people. So these institutions are the most fundamental and fortunate institutions of any country who directly has contact with youth. So which is the best part of the life. And it has a lot of aspiration, imagination, enthusiasm, and it has the promise for the future of any country. The student is one who has a desire to learn, nothing else. So if there is inquisitiveness in a young student, that is a student. So science is something which we actually need for everyday life. If we look around ourselves, we see electric light, computer, television, everything that we see, that we use, is actually given by science. And science defines the course of development through technology. And science also treats everybody equal. And science doesn't have any boundary of institution, country, but it actually spreads all over the world. So if we belong to science, we have a much broader frame of mind and much broader concept of the humanity and society. So we think through science, so it's everybody's benefit. And through a good education program, we actually not only try to give the students, young students, a good training, but through them, we actually see our own future of the country. And through research, we enrich education. So research is very important component of science, because through research, new things come. And anything new has a promise, has a hope. And society is something which includes us, but the society has an expectation from us. Expectation because being public funded institutes, we are fortunate to have funding of the society. And the society's expectation from us is always to give a better environment, better treatment for everybody in the society. So students are actually our angels who go out and spread our message to the society. So if we can inspire students to do science and research, then they bring development to society and to the life of every individual in the society. So it is a continuous, network system. We are not isolated from a society. So can you see this slide? Yeah.
Yes, sir.
Okay. So what is important in life, whether we do science, literature, or economics, or music, or art, or sports, the final goal in life is everybody wants happiness in life. So we must see that the students and ourselves, we all of us, we never compromise this component of life. You can see here myself and my wife, and with my granddaughter, Prachi, and we have got 2 jackfruits, which actually, is grown from the tree which is present in our garden in JNU campus. And it so happens that this tree was planted by my mother when she visited JNU. And since she is no more, this plant and this fruit has a memory. So happiness can be very small things in life, but each person is entitled to happiness. Every individual's life needs happiness and must get happiness. So that can only happen if we have a feeling of sharing with each other. So I would request students, whatever you do in your life, never compromise your happiness. Make sure that you do justice to yourself so that you are happy at the end. Can you see this slide? Yeah. Hello.
The slides, they are not visible. They are visible in the side panel only, sir.
Sir,
Slide.
it is only the second slide and it is not moving. We are still, yes, we can see
now
now
we can
Gandhi
see.
ji. Yes,
Yes. So when you click on the slide, then only it is visible in the center.
Okay,
Otherwise, we are
okay,
viewing
okay.
in the side panel only. Yes, sir.
Okay, did you see this slide?
Yeah.
Now we saw it, sir.
Okay, so this is what I described. You see
Yes, sir. Yes.
myself, my wife, and my granddaughter, Prachi, and we have 2 jackfruits, which is from our garden. So this is a little happiness. I just wanted to show you the picture. So, okay, I have to probably click the slides one by one so that you can see it in the larger frame.
Yes, sir. Yes.
Okay, I'll do that. Don't worry. So can you see this slide?
Yes, sir. Yes.
Oh, so this is the picture of Gandhi ji. So I want to show this as an example to young students that there is always an opportunity or a moment of transformation in life. So transformation in our own life, and through our life, at least as teachers, we are also fortunate, or we are expected to transform the lives of students. So, because through our students, we live and communicate with the society. So, as you can see on the left, it is written first his name, M.K. Gandhi. Then, through his work, he became Gandhiji. Then, people called him Bapu. Then, at the end, we regard him as the father of the nation. So, this person has given an example of life through transformation and sacrifice, and therefore he has given a value system to the society. So much so that the famous scientist Einstein once said that a time will come, people may not believe that a person like this, with flesh and blood, actually lived on this earth. So, transformation is in life is like a catalyst to make changes. So only, of course, few can do that, like what Gandhiji did, but few catalysts can actually change the concept of the society. So the next slide, can you see this slide? Yeah?
Yes, sir. Yes, sir.
Okay, so this slide shows you flowers. So flowers are very beautiful. The moment you see flowers, you feel the sense of beauty. And also, many actually try to draw that. So, because this is a piece of art. So, an artist gets inspiration from flowers, from its color, from its shape and its fragrance. So, artist will draw flowers. But if you look at a poet gets inspiration and writes poetry from the flower. If you look at a science or a scientist, the scientist looks at the science of the flower. Flower is the element which brings you the fruit and maintains the biodiversity in the nature. So flower is an important component of nature. So every student or a young boy or girl is like a flower, which is beautiful and has its future story in it and has fragrance to share with others and spreads a sense of love, which is a higher human quality. Along with that, we also gather knowledge. So if students of botany and teachers of botany would know much more than me about the knowledge on how flowers give us, and once we remember that, that becomes our wisdom. And when these things assimilate in the social culture, that brings us the philosophy. So just like a flower, everything is integrated in one thing. You can see here, we need for social development integration of knowledge, imagination, creativity, contribution, and compassion. So all these components are important, which is essentially required for educational institutions. And a teacher is one personification in whom the students see all these components. So we are very privileged individuals of the society, and all these value systems are important for social development, whether we do science or literature or industry or any other thing. So if you see the next slide, can you see this slide? Yeah?
Yes, sir.
Okay. So this slide says about friendship. So friendship is, yeah. So friendship is a very important component of all through our life and very important for students. So you need to develop your friendship so that you can share your feelings, share your ideas, discuss critically about your ideas, and also get ideas of others. And since in a country like India, we have different language and culture. If we have friendship, we learn a lot of things from others. Somebody is from North India, somebody is from South India, somebody is from Gujarat, Rajasthan, somebody may be from Bengal, Odisha. So we have different culture. So friendship is a relationship which allows us to share and learn more. So pursuit of knowledge is never done in isolation. We can cultivate our own knowledge in isolation, but to share it with others is an important component of this. So never lose your friendship, make good friends. So can you see this slide now?
Yes, sir.
Okay. So what you see is a bougainvillea plant with a lot of flowers, and this is a peak summer time in JNU campus when this flower comes. So this gives us a message that blooming in harsh conditions, when the temperature is very high, there is no water, and this plant learns how to give its best in most difficult conditions. So this gives us the message that for science research, education, or to do anything in the society, first component is we must have confidence in ourselves. So each student must be confident that, yes, I can do something. Yes, we can do something. So that is a very important learning that all of us get when we are on JNU campus. So nobody is actually, anything less than another person. If you try, you can do it. That value system has to be given to the students. So that is very important that they should, otherwise they become, discouraged, disappointed, and sometimes depressed. So that is not a good thing. So can you see this slide? Yeah?
Yes, sir. Yes.
Okay. So here I saw two of my teachers. On the left side, you see Professor M.S. Kanungo, with whom I studied my MSc and did PhD at the Zoology Department, Biochemistry Lab of Banaras Hindu University. He started the research on biology of aging way back in 1960s in India. The first person to think of to do research on aging. See, he established a very good department at BHU, and all of us benefited. His students have spread all over the world. So we learned biochemistry, molecular biology, and he also established another institute in Bhubaneswar, which is Institute of Life Sciences, which is a Department of Biotechnology Institute, which is very good institute now. So it's important to recognize, be grateful to our teachers, because teachers are the main persons who bring change in the society through their students. And in science, it is very important to have good teacher-student relationship. On the right-hand side, you see Professor Charles Wiseman, under whom I learned most of my molecular biology after I did PhD in BHU. So he studied many things and contributed many important things to molecular biology, and was one of the first to establish a biotechnology industry company of the world called Biogen. He discovered interferon genes, then prion gene, and many other things. So he's an inspiring teacher and a mentor for me. So we must have the sense of gratitude to our teachers, and teachers must, we all should try to rise to certain level so that we can contribute to our students. So teachers are very important component of education and science and research. So next I will little bit come to the subject matter. First with molecular biology. Can you see this slide? Yeah?
Yes, yes, yes.
Okay. So what you see here is what we call is the central dogma of molecular biology. How information flows from genes to protein inside cells. So you have DNA here. DNA can make itself through DNA replication. DNA can make RNA through transcription, and RNA can make protein through translation by using ribosomes. And for DNA replication, we need DNA polymerase enzyme. And for synthesizing RNA from DNA, we need RNA polymerase by transcription. Then some viruses have RNA genome. They replicate their RNA genome through an enzyme which is like RNA polymerase, but slightly different. So it uses RNA as a template and synthesizes RNA to copy it. So that's called RNA replicase. Then RNA can synthesize proteins, so that's translation. So we have one enzyme which retroviruses have, reverse transcriptase, which can make DNA from RNA. So the opposite of transcription. Now, so far, there is no knowledge if we can make RNA from protein. So that's a area maybe for future research. So as you see here, DNA and proteins do all the most of the functions in the cells. And in addition to genetics, we have a change the genetic outcome by histone modification, DNA methylation, those things. So can you still see the slide? Yeah?
Yes, sir.
Okay. So if you see, there are some numbers I have written here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So these are 10, on this slide, 10 Nobel Prizes have been given. So you can see on the DNA, first one was DNA as the genetic material. Second was DNA structure. Then the third was genetic code. So number 3 here, which is written near translation. Then fourth was DNA polymerase. And then fifth is discovery of reverse transcriptase. Sixth is splicing, split genes. Then seventh is again PCR of DNA, that we can do amplify DNA in vitro by using PCR technique. Then 8th is ribozyme RNA enzyme, and 9 is RNA polymerase structure, and then 10 is structure of ribosome. So you see here the fourth prize on DNA polymerase here was by for Arthur Kornberg, and the 9th prize listed here, RNA polymerase, is by his son, Roger Kornberg. So father and son got 2 Nobel Prizes. So this is fundamental of biology. So any branch of biology must have this knowledge to begin with. So simple fundamental discoveries are important in science and research because they give us new knowledge, show us the path for new discoveries. So if we go to the next one, can you see this next slide?
Yes, sir.
Okay. So this slide shows you chromosomes. On the left side, we have chromosomes of maize, the corn, and the right side, we have chromosomes of human cell. So here you have, basically 7 chromosomes shown here, 7 pairs. And you see these dark spots here, they are actually heterochromatin, condensed chromatin, and which contains repetitive DNA. And if you see in the human chromosome, human cell, you have these dark bands.
Audience, please mute your mics.
ये इधर कंफर्टेबल है क्या? हाँ ठीक है कंफर्टेबल है क्या बच्चे सारे? हाँ ठीक है ठीक है थोड़ा दूर हटो.
Sorry for the disturbance, sir.
Sir, please unmute yourself now. I've muted all.
Oh, okay. So can you hear me?
Yes, sir. Yes, sir.
Yes.
Okay. So on the right side, you see the G bending, human chromosomes. the dark bands, they represent actually repetitive DNA. So one of the fundamental question is, in the chromosomes, why we have so much of repetitive DNA? And if we see, okay, so let me show the next slide. So this is, can you see the slide? Yeah.
Yes,
Yes,
sir.
sir.
Okay.
Yes, sir.
So this is another way of staining human chromosomes. So you can see the spots here, the yellow spots, and most of them are actually repetitive DNA, human chromosomes. So now if you see the next slide, can you see this slide?
Yes, sir.
Right. So the chromosomes are in blue, then you have two red dots at the ends. So these red dots are actually telomere. And you can see every chromosome pair at the end, both ends, have actually two dots, the telomeres stand here. So telomeres are also repetitive DNA at the end of the chromosome. Now, if you see this slide, here, this slide, can you see this slide?
Yes,
Yeah.
sir.
So if you see some chromosomes, they have only one, like this chromosome, you have only one yellow dot. The other one doesn't have that yellow dot. Like here also, one end doesn't have. So here also one end doesn't have, one end doesn't have. This is because these chromosomes are from the cells of a old person. So in old days, you can see here in this chromosome, there are faint yellow dots. So telomeres are decreased, they are lost. Like here you can see on this chromosome, one end it is there, the other end it's not there. So one of the things which happens during aging or when we become old, our chromosomes lose some of their telomeres. And due to that, a lot of things happen. I'm not going to all the details here, but just trying to tell you that we can actually capture the genetic or genomic changes which occur in cells like this. So this I just showed you. Now the next one, if you see the slide, can you see this slide?
Yes,
Yeah?
sir. Yes, sir.
So this is the three blue thing that you see is three nuclei, nucleus of a cell. And you can see on the top, you have this pink color dots. They are telomere sequences in the chromosomes. And that is a normal cell. And you can see in the B, there is a cell, you have more pink color staining due to more telomeric sequences. This is a cancer cell. Okay. Now at the end, you see C, you have even more pink color stains. So telomeres increase in
I think there is a connectivity problem with And Dr. Rath.
Certain technical issue at his side.
Maheshwari, madam, you can ask sir whether he's facing any technical problem.
He has joined again, ma'am.
Sir, unmute yourself.
Sir, please unmute yourself.
मैं फोन करती हूँ.
आप म्यूट हो जाइए फिर उसके.
Yeah.
हाँ.
Yes, sir.
Okay.
Can you see the slide?
No, sir.
No, sir.
Okay. Just a moment, please. So because it got disconnected somehow, so maybe I have to possibly share again. Or... Can you see?
Yes, sir.
Okay. So this is a problem with can you see the slide? Yeah?
Yes, sir.
Okay. So what you see here is Barbara McClintock. On this black and white picture is, when she did the work, and the colored picture on the right is when she got the Nobel Prize in 1983. She discovered transposons, mobile DNA, or the concept that genes or DNA sequences can jump from one site of the chromosome to another site. So you see a lot of maize here with colored dots, colored kernels. So these are colored due to transposable DNA, transposons, which have jumped at different points of time. So Barbara McClintock did her research and finding of mobile DNA, jumping genes, jumping DNA around 1930 to 1950, and she got Nobel Prize in 1983, so almost 50 years after. And what she had discovered in case of maize, we found the same information in the human genome in 2001 when the whole genome was sequenced. That human genome has got 50% repetitive DNA and has got 45% transposons. So you can say she was 69 years ahead of time. So she mentioned once that imagination is more powerful than knowledge. So we get some knowledge through our experimentation, science, research, but based on that, we have to apply mind, imagine further. So that's a very important component of research in science education. So can you see this slide?
Yes, sir.
Okay, I have to give some concrete examples that did we inspire some students, or we are just telling others that you should inspire. So I will give few examples of my own PhD students. You see the picture of Deepak here, who did his PhD between 2006 to 12, and he actually analyzed some jumping gene or mobile genetic element or transposon in the brain of the rat. And now, through his work, Deepak got his position in New York, in Chicago and New York, and he's doing excellent cancer research in a very reputed institute in New York now. So Deepak came from Azamgarh in UP, came to JNU to do PhD, And through PhD, he transformed himself. Now I can say I am proud of him because he is doing cutting edge cancer research in one of the best places in USA, in a very advanced country. So for students, it is very important that it is not necessary that you only remain in one place. In science and research, wherever you are, if your work is good, you can contribute to the entire world. So one example I just want to say. So can you see this slide?
Yes. Yes, sir.
So on the rightmost side, if you see, is blue color staining is because of expression of a transposable sequence or transposon in the brain of the rat. So the top one is, just a moment. So the top one is cortex part of the brain. Second is hippocampus part of the brain. So hippocampus part of the brain is responsible for learning and memory and adult neurogenesis. Then the third part, lower part is from the cerebellum. All these blue dots that you see is neurons expressing transposons. So as of today, we do not know completely everything about why these transposons, which are mobile genetic elements, are expressed so much in the brain, what function they have in the brain. So we are looking into that aspect. So this is what part of the Deepak and subsequent students' work also. So can you see this slide?
Yes, sir.
Okay, another student, Suklin. She did her PhD with us with me in 2000, from 2009 to 15. She worked on long non-coding RNAs. So the messenger RNA makes protein, but there are non-coding long RNAs who do not make proteins, but they regulate most of the functions in cells. That's what is also one of our research interests. So Suklin is now in a medical center in Pittsburgh, and she is doing extremely good work on neurodegenerative diseases in human brain, in human patients. So I am also proud of her that she came from Jammu and joined her PhD, and through her work, she transformed herself. So research and education has enormous power to change, yourself, and to change through your work, to change the world. So this is another example. So you can see one piece of work, what she did. I don't want to go into all the details. Can you see this slide?
Yes, sir.
Okay, on the right hand side, you see red color cells and the center is blue. So blue is the nucleus and red is the RNA, long non-coding RNA expressed in the cytoplasm of the cell. And the left side, you see these cells, which are actually neurons isolated from the brain hippocampus of the rat brain. Okay, so this is one picture of research data. I just wanted to show as an example how it looks like to get a feeling. So can you see this slide?
Yes, sir.
Okay. So this picture, what you see is different type of cells in the testis, mammalian testis. So testis makes sperms, male gametes, and testis makes billions of sperms. So that actually gives genetic variation to the species. So if a species has to improve itself through evolution, then it needs genetic variation as a substance for evolution to act upon. So this is what you see. Ajay came from Rajasthan, and he studied, he just submitted his PhD thesis. He studied how these long non-coding RNAs are working in different type of cells in the testes and what happens to them in aging. So he has published now 2 papers in top ranking science journals of the world. So I am also very proud of him. So Ajay also, through his research and education, transformed himself and gave new knowledge to the world. So I'll give one more type of work. Can you see this slide?
Yes, sir.
So this is Jiten. Jiten did work on bone marrow stem cells, what you see here on bone. And inside the hole, there is marrow which contains cells, and from that we get mesenchymal stem cells. So Jiten is now teaching in Shivaji College of Delhi University. He did excellent work on mesenchymal stem cells, and he also transformed himself and now transforming lives of many BSc students in zoology department of Shivaji College, Delhi University. So you can see a little bit work of stem cells just to show some pictures, not to bore you with too much of science. So you can see this is a colony. Can you see this slide? Yeah?
Yes, sir. Yes, sir.
Okay. These are mesenchymal stem cells from mouse bone marrow. And individual cells you can see here, how they look under microscope. And these cells grow like a colony with millions of cells. And these can work like stem cells for repair of the tissues when the tissue is damaged. So I'll show you 2 examples of that. One, you see this, when we collected these mesenchymal stem cells, Jiten differentiated them in lab. Can you see this slide? Yeah?
Yes, sir. Yes, sir.
Okay. On the top side, you see top panel, mesenchymal stem cells get differentiated into adipocytes. The fat globules that you see, red color, this is one adipocyte cell. In the middle, you see mesenchymal stem cells can be differentiated into bone cells. So you can make bone in the lab, bone cells. So this is single osteocyte you see. On the lower side, you see mesenchymal stem cells getting converted to chondrocytes or cartilage cells. Here is the red stained cartilage cell. So these cells can be used for regenerative medicine. So another student, Naseem, I think he came from some part of Western UP, and he also worked on bone marrow stem cells. And you can see here a bone on the left and a damaged marrow of the bone, the whole. Can you see this slide? Yeah?
Yes, sir.
Okay. After radiation, and on the right side, after transplantation of stem cells, this damage is repaired. So Naseem also did excellent research on bone marrow stem cells, and he published his work. So Naseem is now working in Texas as a scientist, independent scientist, and doing very good work on stem cells. So you can see that slide little bit in details. So this is the control, all 3 of them, bone marrow, radiation, the middle one, the part of the middle cells are lost. So there is hole in the marrow, and top is transplantation. When you give new stem cells, then you can repair the bone marrow. So that is a regenerative medicine approach, which Naseem did. So these are few examples of students who did their research with us and actually transformed and contributed. Can you see this slide?
Yes, sir.
So since we work on aging, These are some of the books where we have described all aspects of aging, all molecular aspects, because aging is becoming a big problem all over the world. And due to old age, we are also getting old age associated diseases. So these books are there. Anybody interested can, get into it. So what you see here is, can you see this slide?
Yes, sir.
Okay. So these are few students: Rasmi, Deepak, Sukleen, Manish, Jiten, Yashin, Jyoti, and Sandeep. All of them, through their research, actually transformed their lives, and they are very actively contributing to the society now. So that's a concrete example of science, education, and research, how it can help in changing and contributing to the society. And right now, the two students who are working, Ajay has finished. So this is Ajay, this is Anita. So they are like 19th and 20th PhD students with me. So the idea is, if you can generate 20 good PhD students, they can generate another 200 students and can contribute to the society. So university, through their education and research, can change the lives of students and can build good society. Can you see this slide?
Yes, sir.
So this is our JNU campus, which is very natural, and you see a lot of trees and very good, and which doesn't look like a man-made garden, but is a natural forest. So this is very inspiring to us. And I look at it as a picture or piece of art, because I will conclude with what a great artist, Pablo Picasso, who through his art, paintings, changed the society and human thinking. He said that art washes away from the soul the dust of life. So with science, we need art also. And scientific research, the data, actually is, each one is like a piece of art. So we need both, science and art. So with these words, I think I thank you very much for your invitation and patient listening. Thank you very much. If you want any discussion, we can have it. And I think I have already consumed my time. And I see Dr. Mishra is there already logged in, probably for the next lecture. So thank you very much.
So I would like to ask questions.
Yeah.
Yeah, there are two questions. First, when we are visualizing the slides of cancerous cells, you have seen that there are more telomeric, the pink color, the stain. That means there are more telomeres. Yes, this one, this slides.
Yeah.
Yeah, can you explain the normal they are having come and telomeres? Telomeres are actually responsible for stability of cell.
Yes. So this is a normal cell, the top one.
Yes.
The middle one is a cancer cell, which you see more telomere. The pink dots have increased, right?
Yeah.
The telomeric sequences in cancer cells, they are not only at the end of the chromosome, but they kind of spread into the chromosome also. This is abnormal multiplication of telomere. And this lowermost is a more aggressive cancer cell, which has
that means they are the marker ones.
They can be marker, they can be target, they can be a lot of things. So you have more telomere. So this is one example how the genome is changing in cancer, right?
Okay. Okay. Another question, there are just in the we have seen that gone through that there are near about 40% of transposons, the jumping genes in humans. So are there any
Yes.
examples how they have affected our either clinical or any such of the systems, because they can be get inserted in any of the coding sequences and then
Exactly. You are absolutely correct. So there are two types of information if you see literature. One, that they are transcribed and sometimes they can disrupt genes. And that is one way there are some diseases like thalassemia and others. And during normal expression of these kind of transposons, they are also involved in normal physiology of the cells. So they're not always bad, but when there is abnormal expression or abnormal transposition, then they can cause disease or mutation. Yeah.
yeah. Thank you.
Thank you so much, sir, for sharing your amazing work and work of your scholars and their success stories with us. I hope all the audience, students who have joined with us, they must have learned how to transform their lives using the tool of science and then serve the society as well. And you also shared a beautiful pictorial presentation of the simple joys of life and how they add value to our lives. Thank you, sir.
Thank you very much.
Thank you, sir.
Thank you very much. Bye-bye. So can I log off now?
Yeah, yes, thank you, sir, for such an enlightening lecture you have seen. You have all either starting from central dogma to the expression and how it helps in aging to the transposability. It was very interesting lecture for the students and for us also. So thank you for joining us.
Thank you very much. Thank you.
So now we can move on to our third technical session. And we are privileged to have Dr. Dinesh Mishra, sir, with us, an ophthalmologist and president of Andhashraddha Nirmulan Samiti, Raipur, Chhattisgarh. Sir will be speaking about scientific temperament and social superstitions. I request Dr. Renu Maheshwari ma'am to introduce our guest speaker.
Thank you, Dipali. Can I audible?
Yes, ma'am.
Okay, thank you. Good afternoon to all. रायपुर शहर के वेल नोन ऑप्थेल्मोलॉजिस्ट व सामाजिक कार्यकर्ता जो समिति रायपुर छत्तीसगढ़ के प्रेसिडेंट हैं, डॉक्टर दिनेश मिश्रा 1995 से सामाजिक कार्यों में संलग्न है, जो लगातार वैज्ञानिक दृष्टिकोण के विकास एवं सामाजिक कुरीतियों के निर्मूलन के लिए जग जागरण अभियान छत्तीसगढ़ सहित देश के अन्य राज्य असम, राजस्थान, उत्तर प्रदेश, उड़ीसा, आंध्र प्रदेश, कर्नाटक, बिहार, झारखंड तथा महाराष्ट्र के अनेक स्थानों में इस विषय वैज्ञानिक जागरूकता के विकास के लिए आपके द्वारा लगभग 2200 से भी अधिक जन सभाएं आयोजित की गई हैं। केंद्रीय विज्ञान एवं प्रौद्योगिकी संचार परिषद एवं प्रौद्योगिकी मंत्रालय द्वारा भी आपकी भागीदारी रही है। राष्ट्रीय विज्ञान संचार सम्मेलन वाराणसी, दिल्ली में आपके द्वारा सामाजिक कुरीतियों के ऊपर शोध पत्र प्रस्तुत किए गए हैं। सूर्य ग्रहण एवं चंद्रग्रहण जैसे विज्ञान प्रसार के आयोजनों में सक्रिय भागीदारी रही है आपकी, जहां आपने इन विषयों पर व्याख्यान दिए हैं। आपको देश भर से चमत्कारों के वैज्ञानिक व्याख्या, दैट इज साइंटिफिक एक्सप्लेनेशन ऑफ की कार्यशाला में मास्टर रिसोर्स पर्सन तथा विशेष विशेषज्ञ के रूप में आमंत्रित। अंधविश्वास के कारण जादू टोने के संदेह में महिला प्रताड़ना के विरोध में कोई नारी डायन नहीं अभियान, जिसमें आप लगातार ग्रामीण अंचल के दौरे, पीड़ितों से मुलाकात, पुनर्वास का कार्य, सामाजिक बहिष्कार की घटनाओं के विरोध में अभियान एवं जगजागरण पर कार्य लगातार करते आ रहे हैं। समाज की इन कुरीतियों पर साइंटिफिक एक्सप्लेनेशन पर आप विगत 28 वर्षों से लेखन कर रहे हैं, जो पत्र पत्रिकाओं में नियमित कॉलम के रूप में प्रकाशित हो रहे हैं। अब तक इन्हीं विषयों पर आपके द्वारा 7 किताबें लिखी गई हैं, जो राज्य शासन एवं साक्षरता मिशन द्वारा अनुशंसित तथा प्रकाशित की गई हैं। केवल देश में ही नहीं, समाज की कुरीतियों, विशेष रूप से अंधविश्वास पर आपको सन 2014 में ब्रसेल, बेल्जियम में तथा 2017 में यूनाइटेड नेशन द्वारा जिनेवा में व्याख्यान के लिए आमंत्रित किया गया है। समाज में आपके द्वारा वैज्ञानिक दृष्टिकोण से नई दिशा प्रदान करने के लिए कई सम्मानों से भी नवाजा गया है। यू आर ऑनर्ड बाय Honorable Governor of UP, Shri Motilal Ji Vora in 1995, 8th Red and Bravery Award for work against superstition and public awareness movement presented by Mrs. Sheila Dikshit, Chief Minister of Delhi on May 1999. You are honored by Governor of MP at Bhopal in 1999 for social services. Dr. Dinesh Mishra is honored by Indian Medical Association MP State branch in annual state conference at Bilaspur in 1999. Has honored on Doctor's Day for social work on July 1999. Government of as a Chhattisgarh Alankaran on Establishment Day of Chhattisgarh State. President of India Honorable Dr. APJ Abdul Kalam facilitated Dr. Mishra for his community and social services in November 2000. Government of Chhattisgarh honored as Chhattisgarh Alankaran. President, Department of Science and Technology, Government of India, presented National Award in 2007 for commendable effort in science and technology, communication, and promoting scientific temper in the country. Chhattisgarh State Women Commission honored for community and social services in 2007. Speaker of Chhattisgarh Assembly, Honorable P.P. Pandey, facilitated Dr. Mishra, also honored by Nehru Yuvak Kendra for community and social services. I welcome you, sir, for your talk on scientific temper superstition. Welcome to sir. डॉक्टर दिनेश मिश्रा सर आप ज्वाइन कर लिए ना, प्लीज ज्वाइन अस।
जी आप मेरी आवाज सुन पा रही हैं सब ठीक से?
जी जी सर।
जी 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 धन्यवाद। मैं इस साइंटिफिक सेशन में आप सभी का हार्दिक अभिनंदन करता हूं। और जो हमारा आज का जो टॉपिक है साइंटिफिक टेम्परामेंट और एंड सोशल सुपरस्टिशन, जैसा कि माहौल चल रहा है पूरे देश में, इसमें साइंटिफिक टेम्परामेंट जो है इसको जगाने की जरूरत बहुत अच्छे से महसूस की जा रही है और बहुत तेजी से महसूस की जा रही है। यहाँ हम सब विज्ञान के विद्यार्थी जुड़े हुए हैं, इसमें साइंस जो लोग हैं, इसलिए मैं बहुत शुरुआत से बातें करना चाहूंगा। जो साइंस की बात हम करते हैं कि साइंस जो है, साइंस शब्द जो है, इसकी जो उत्पत्ति हुई थी, एक बहुत बेसिक बात करना चाहूंगा मैं। लैटिन शब्द सेंटिया से हुई थी, जिसका अर्थ था ज्ञान, नॉलेज या जानना, टू नो। अर्थात कि हमारे इस जगत में जो कुछ भी घटित हो रहा है उसका क्रमबद्ध एवं सिस्टमेटिक नॉलेज ही साइंस कहलाता है। और जब हम इस प्रकृति में, नेचर में उपस्थित अलग अलग वस्तुओं की प्रकृति, उनके व्यवहार जैसे गुण इत्यादि का अध्ययन करते हैं तो उसे हम विज्ञान कहते हैं। और इनके क्रमबद्ध तरीके से अध्ययन को किया जाता है, तथ्य इकट्ठे किए जाते हैं, इसके आधार पर किसी वस्तु के गुण एवं प्रकृति का पता लगाया जाता है। मुझे लगता है कि जो वास्तविकता का ज्ञान है हम सब के बीच में, वही विज्ञान है। जैसे कुछ प्राकृतिक सत्य जो है वो हमेशा से सत्य हैं, पर हम उन्हें पहले से मानते थे, लेकिन उसकी जो पूरी सच्चाई है, बहुत सारी घटनाओं के बारे में, प्राकृतिक घटनाओं के बारे में, तथाकथित चमत्कारों के बारे में, उसकी संपूर्ण सच्चाई से हमें विज्ञान ने अवगत कराया है। जैसे पानी उबलता है, ये हम सभी को पता था, बरसात सदियों से पता था, लेकिन पानी 100 डिग्री सेंटीग्रेड पर उबलता है, यह विज्ञान ने बताया। पहले मानव जाति मानती थी कि पृथ्वी स्थिर है, जबकि सूर्य पृथ्वी की परिक्रमा करता है, ये लोग मानते थे। लेकिन बाद में विज्ञान ने लोगों को बताया कि जो पृथ्वी है वो सूर्य की परिक्रमा करती है। और यह जो वैज्ञानिक बातें लोगों को धीरे धीरे समझ में आई हैं, उससे पूरे संसार में एक साइंटिफिक टेम्परामेंट बनने की शुरुआत होती है। और वैज्ञानिक दृष्टिकोण क्या होता है, इसकी आवश्यकता क्यों है? जैसे कि मैंने पहले कहा, वैज्ञानिक दृष्टिकोण, साइंटिफिक टेम्परामेंट का अर्थ ही है किसी कही सुनी बात को आंख मूंद कर स्वीकार ना करे, उसको तर्कों एवं प्रयोग की कसौटी पर परख कर ही स्वीकार करना। आज आप देख रहे हैं व्हाट्सएप के झूठे मैसेज, फर्जी बाबाओं के झांसे में आकर लूटने वाले, ठगों की फर्जी काल से अपना पैसा लुटाने वाले लोग, झटपट अमीर बन जाने के झांसे में आकर अपनी मेहनत की कमाई लुटाने वाली जैसी घटनाओं से बचे रहने के लिए हम सब में वैज्ञानिक दृष्टिकोण बनाना और उसका विकसित करना बहुत अनिवार्य हो गया है। विज्ञान जो है ये एक सीखने की प्रक्रिया है, और जो वैज्ञानिक दृष्टिकोण है ये हमको अपने परिवेश को समझने और परखने का अवसर देता है। वैज्ञानिक दृष्टिकोण हमारे अंदर एक अन्वेषण को, एक जिज्ञासा की प्रवृत्ति को विकसित करता है और विवेकपूर्ण निर्णय लेने में सहायता करता है। वैज्ञानिक दृष्टिकोण की शर्त यह है कि बिना किसी प्रमाण के किसी भी बात पर विश्वास ना करना या उपस्थित प्रमाण के अनुसार किसी बात पर भरोसा करना, चर्चा करना, आपस में तर्क करना, विश्लेषण करना, यह वैज्ञानिक दृष्टिकोण का एक महत्वपूर्ण अंग है। और अंधविश्वास क्या है? शाब्दिक अर्थों में जाए तो अंधविश्वास का अर्थ हुआ किसी भी बात पर आंख मूंद कर भरोसा कर लेना या बिना समझे विश्वास करना। यह कहा जा सकता है कि किसी विषय को अगर बिना जाने बूझे हम भरोसा करते हैं, यह अंधविश्वास है। इसके कारण क्या क्या हो सकते हैं अंधविश्वास के? सामाजिक सोच, कई बार जो परंपरागत सोच होती है ये आगे आपकी तर्कशीलता को रोकती है। अशिक्षा, अभी शिक्षा की दर बढ़ी है, लोग अधिक शिक्षित हुए हैं, लेकिन उसके बाद भी बहुत सारे लोग हम सिर्फ साक्षर कर पाए हैं, उनको हम व्यवहारिक रूप से, वैज्ञानिक रूप से उनको हम शिक्षित नहीं कर पाए हैं। स्वास्थ्य सुविधाओं की जो कमी होती है वह भी एक अंधविश्वास के बढ़ने का कारण है। सही मार्गदर्शन एवं जानकारी का अभाव, सामाजिक मामलों में, चिकित्सा के मामलों में सही मार्गदर्शन ना मिलना, सही जानकारी ना मिलना, यह भी एक अंधविश्वासों का कारण है। वैज्ञानिक दृष्टिकोण की कमी, जिस बात की जो मैंने कहा, एक जो हमारे बीच में छोटा बच्चा होता है वो सबसे बड़ा वैज्ञानिक होता है, क्योंकि वो हर घटना के पीछे कारण जानने की कोशिश करता है और वह जानना चाहता है कि बिजली क्यों जल रही है, बिजली कैसे बंद होती है, कैसे चालू होता है, कैसे बंद होता है, ग्रहण कैसे होता है, इत्यादि इतनी प्राकृतिक घटनाएं होती हैं जो देखता है, जो सामने की घटनाएं होती हैं उसके पीछे वो कारण जानने की कोशिश करता है, वो तर्क करता है, और यह वैज्ञानिक दृष्टिकोण बढ़ने का कारण है। लेकिन जैसे जैसे बच्चा बड़ा होने लगता है, कई बार वो उन्हीं पुरानी बातों पर मानने लगता है। टीवी पे आ रहे लगातार विज्ञापनों, बाबाओं के विज्ञापन है, उनके जो प्रचार के माध्यम है, उनके झांसे मानने लगता है। आर्थिक समस्या भी एक बहुत बड़ा कारण है अंधविश्वास का। हमारे देश में अभी भी बहुत सारे हिस्से ऐसे हैं जहां पर लोग जो आर्थिक रूप से समृद्ध नहीं हैं, उनके पास चिकित्सा की सुविधा नहीं है, उनके पास मार्गदर्शन लेने के पास जाने का समय नहीं है, आर्थिक रूप से वो परेशान है। इस कारण भी लोग बहुत सारे अंधविश्वासों को मानते हैं कि उसको कर लेने से वो सफल हो जाएंगे या वो ठीक हो जाएंगे। अंधविश्वास का हमने वर्गीकरण भी किया है कि किस प्रकार के अंधविश्वास होते हैं। एक तो जैसा मैंने कहा कि सामाजिक जो अंधविश्वास होते हैं, इसके अंतर्गत हम लेते हैं सूर्य ग्रहण जैसी घटनाएं जो हैं, चंद्र ग्रहण जैसी घटनाएं हैं। हम अपने बच्चों को प्राइमरी कक्षा से पढ़ाते हैं कि ग्रहण क्यों होता है, छाया पड़ने से ग्रहण होता है, सूर्य पृथ्वी बीच चंद्रमा के आने से होता है। यह सब बातें हम बच्चों को अपनी प्राइमरी कक्षा से पढ़ाते हैं। लेकिन आप देखिए, जब भी देश में या पूरे संसार में हर टीवी के चैनल पर एक बाबा आकर बैठता है, लोगों को बताने लगता है कि ग्रहण क्यों है और इस समय हमें क्यों बाहर नहीं निकलना चाहिए, हमें इसमें क्या करना है, क्या नहीं करना चाहिए। जबकि जो हमारा डिपार्टमेंट ऑफ साइंस टेक्नोलॉजी है, ये बरसों से लोगों को जानकारी दे रहा है कि आपको भाई इन सब खगोलीय घटना है, आप इसको सुरक्षित रह करके देखिए, आप किसी भी अंधविश्वास पे ना पड़े, किसी प्रकार की रोक नहीं है, सिर्फ अगर आप सुरक्षित ढंग से देखिए तो आपको नुकसान नहीं करेगा। लेकिन इसके बाद भी आज भी बहुत सारे लोग हैं वो ऐसी बातों पर, अवैज्ञानिक बातों पर भरोसा करते हैं, उनके झांसे में आते हैं। जो प्राकृतिक घटनाएं हैं, सूर्य ग्रहण के साथ में बाढ़ है, अकाल है, भूकंप है, इससे बहुत सारे अंधविश्वास जुड़े हैं और मीडिया कई बार ऐसे अंधविश्वासों को बढ़ावा देता है। जादू टोने की जो मान्यता होती है, वैज्ञानिक दृष्टिकोण के अंतर्गत हमको एक बात कहना चाहिए, जो जादू टोने की जो मान्यता है, टोनही की मान्यता है, डायन की मान्यता है, यह एक ऐसा अंधविश्वास है जिसने लाखों परिवारों को अपने संकट में डालकर रखा है। साक्षर होने के बाद भी बहुत सारे लोग वो ऐसी बातों पर भरोसा करते हैं कि इस प्रकार का कोई ब्लैक मैजिक होता है, कोई जादू टोना होता है। और आपको जान के आश्चर्य होगा कि हजारों महिलाएं जो हैं वो इस प्रताड़ना की शिकार होती है। उन्हें जादू टोने के शक में डायन कहकर, डाकन कहकर, टोनी कह के प्रताड़ित किया जाता है, मारा पीटाया जाता है। जो नेशनल क्राइम रिकॉर्ड ब्यूरो है, उसके रिकॉर्ड के अनुसार जो है देश के 17 प्रदेश ऐसे हैं जहां पर इस प्रकार की घटनाएं होती हैं, और वैज्ञानिक दृष्टिकोण के अभाव में लोग जो है अंधविश्वासों के शिकार होते हैं और महिलाओं को प्रताड़ित करते हैं। बिहार में ऐसी घटनाएं बहुत सारी होती हैं लगातार। छत्तीसगढ़ में है, महाराष्ट्र है, मध्य प्रदेश है, असम है, राजस्थान है, पंजाब है। लगातार हमारे पास आंकड़े हैं और हम बातचीत करते हैं, काम भी करते हैं। जबकि यह मैं अब इस बात को मैं आपसे फिर से कहना चाहूंगा कि इस प्रकार की मान्यता पर लोगों को भरोसा नहीं करना चाहिए। और अगर हमारे आसपास कोई ऐसा व्यक्ति है जो सब चीजों पर भरोसा करता है, तो हमको उसको कहूंगा तो आप आश्चर्यचकित जाएंगे कि इस सिर्फ अंधविश्वास के कारण जो घटनाएं होती हैं उसमें महिलाओं को किस प्रकार प्रताड़ित किया जाता है। हमने इसमें सर्वेक्षण भी किया था कि इसमें बहुत सारी महिलाओं की हत्याएं हुई हैं, गंभीर रूप से घायल महिलाएं हैं जिनको गर्म सलाख से दागा गया है। अभी एक घटना छत्तीसगढ़ में हुई है, उसके पहले असम में हुई थी। उन्हें आग का घेरा पार करने को कहा जाता है, उनके साथ मारपीट की जाती है डंडे से, इमली के डंडे से, मलमूत्र का सेवन कराया जाता है, अंग भंग करना। एक महिला से मैं मिला था जिसके जीभ काट दी गई थी, उसकी आंख तोड़ दी गई थी। बाल काटना, कुछ लोगों को लगता था कि भाई बाल काटने से वो बच जाएंगे। कुछ स्थान पर सार्वजनिक अपमान भी हुआ है, उन्हें कपड़े उतार कर घुमाया गया है, गाली गलौज की गई है, उनके गांव से बहिष्कार किया गया है, जुर्माना किया गया है। इससे बहुत सारी महिलाओं ने आत्महत्या भी की है। और हमने जब सर्वेक्षण किया तो हमने पाया कि इसमें जो मामले हैं ये करीब 99% जो मामले हैं ये ग्रामीण अंचल के हैं। जब हम शिक्षा की बात करते हैं तो हमको साथ में इस बात की भी आवश्यकता है कि हम जो सामाजिक कुरीतियां हैं उसको भी हम हटाने के लिए आगे बढ़े, उस पर काम करें। और इसके साथ ही जो कुछ अंधविश्वास ऐसे हैं जो आपको बताऊंगा, जो आपको हंसी आएगी, जो आंख फड़कने की बात होती है। इसमें लगातार हम लोगों को बताते हैं कि बी कॉम्प्लेक्स कॉम्प्लेक्स की कमी से फड़कते हैं, लेकिन बहुत से लोग इस बात पे भरोसा करते हैं फड़कने से हो जाएगा। यह जो बात है बहुत से लोग आज भी मानते हैं। जो अंग फड़कने की बात होती है, छींकना, अभी भी बहुत सारे स्थानों पर लोग मानते हैं कि छींकने से अशुभ होता है, जबकि डॉक्टरों का ये मानना है कि जो छींकना है, हमारी नाक में जो म्यूकस होता है, जो हमारे में जो बहुत सारे श्लेष्मा होती हैं, जो बहुत सारे जो छोटे छोटे बाल होते हैं, जो हेयर होते हैं, वो उनको रोकने का काम करते हैं, किसी भी बाहरी गंध को, किसी बाहरी वस्तु को, और उसके लिए हम छींकते हैं। लेकिन इसमें किसी को नुकसान पहुंचाने की बात नहीं हो पाती है। बहुत सारे स्थानों पर लोग ये मानते हैं कि सोने की वस्तु गुम हो जाएगी तो अशुभ हो जाएगा, अंग फड़कने से अशुभ हो जाएगा, कौवा दिखता है उसको लोग मानते हैं अशुभ हो जाएगा। जबकि जो कौवा और इस प्रकार की जो चीजें हमारे पर्यावरण का एक अंग है। और जब भी पहले नगर निगम नहीं थे, म्युनिसिपल्स नहीं थे, गांव में ग्राम पंचायतें नहीं थे, तो वे ऐसी वस्तुओं को साफ करते थे जिससे गांव प्रदूषित होता था। एक अंधविश्वास जो सामान्य सा है, बच्चे को लांगने से उसका कद ना बांधना। एक मैं बहुत सी छोटी छोटी बातों पर जा रहा हूँ। बहुत सारे स्थानों पर लोग मानते हैं ग्रामीण अंचल में किसी बच्चे को अगर लांग दिया जाए तो उसकी ऊंचाई नहीं बढ़ती, जबकि विज्ञान का, चिकित्सा विज्ञान का मानना है और हम सब का मानना है कि किसी बच्चे की जो ऊंचाई है ये इस बात पे निर्भर करती है कि उसके माता पिता की ऊंचाई कैसी है, उस बच्चे का पोषण कैसा है, बच्चा कुपोषित तो नहीं है, इस बात पर निर्भर करती है। किसी के लांगने से उसकी ऊंचाई जो है उस पर असर नहीं होता है। एक अंधविश्वास जो सामान्य रूप से ग्रामीण अंचल में हम देखते हैं, बाहर जाने पर अगर किसी ने टोक दिया तो कहते हैं अशुभ हो जाएगा, जबकि यह हम विज्ञान के विद्यार्थी से जुड़े हैं। हमारे घर में हमको तभी टोका जाता है जब हमारी गलती सुधारने का मौका आता है। जैसे बच्चे परीक्षा देने जाते हैं तो उनकी जो माताएं हैं उनको उस समय टोका करती हैं कि भाई अगर आपने अपने एडमिशन कार्ड गुम गया है, एडमिशन कार्ड नहीं लेकर जा रहे हो, पेन नहीं है, गाड़ी की चाबी नहीं है, तो आप परीक्षा नहीं दे पाओगे। एक अंधविश्वास जो सामान्य सा है, जो सामाजिक अंधविश्वास में आता है, बहुत सारे स्थानों पर लोग मानकर चलते हैं हाथ खुजाने से पैसा मिलना। एक बहुत सामान्य सी एक लोगों में भ्रांति है, जबकि हाथ खुजाने से पैसा जो है, मैं आपको फिर से बताना चाहूंगा कि इसमें सिर्फ डॉक्टरों को मिलता है क्योंकि वो उनका इलाज करते हैं। हाथ खुजाने से किसी व्यक्ति को पैसा नहीं मिलता है। जो दिशा शूल की मान्यता है, आज भी समाचार पत्रों के कॉलम में देखिए बहुत सारे स्थानों पर लिखा रहता है कि आप इस दिन दक्षिण दिशा में मत जाइए, किसी दिन उत्तर दिशा में मत जाइए। लेकिन ये सब जो बातें हैं यह उस समय की बातें हैं जब हमारे पास यातायात के साधन बिल्कुल नहीं थे और लोगों को आने जाने के लिए बहुत कम साधन थे। गांव के चारों तरफ जंगल थे, कोई भी ऐसी घटना हो जाती थी जिसमें लोग दुर्घटना के शिकार होते थे, इसलिए लोगों ने ऐसे नियम बनाए होंगे। लेकिन आज के युग में जब हमारे पास हर दिन चारों दिशाओं में जाने के साधन हर दिन है, तो हमको ऐसी बातों पर भरोसा करने की आवश्यकता नहीं है। एक सामान्य सामाजिक अंधविश्वास है ठोकर लगने को गाली देने का परिणाम मानने की जो बात होती है। बहुत सारे लोग मानकर चलते हैं, लेकिन किसी भी बच्चे को, किसी भी छात्र को ठोकर तभी लगेगी जब वे व्यक्ति यातायात के नियमों का पालन नहीं कर रहा है। उसको सिर्फ किसी के गाली देने से ठोकर नहीं लगेगी। एक ग्रामीण मान्यता है कि अगर किसी की सपने में किसी को मृत्यु देख लिया है तो उसकी उम्र बढ़ती है। इसका भी कोई तार्किक आधार नहीं है। भूत प्रेत की जो मान्यता है और उसकी मुक्ति के लिए जो ताबीज वाली बात होती है, मैं यह आज कहना चाहूंगा कि भूत प्रेत जैसी कोई जो बात है इस दुनिया में नहीं है। बहुत सारे लोग इस बात पर लगातार वाद विवाद भी करते हैं, बहस भी होती है। हम पिछले 25 साल से छत्तीसगढ़ में हम रात में जाते हैं, ग्रामीण अंचल में जाते हैं, शमशान घाट में जाते हैं, और हमें आज तक कभी कोई भूत प्रेत नहीं मिला। और ऐसी घटनाएं जो होती हैं, ऐसी महिलाएं, ऐसी बच्चियां जो एनीमिक हैं, जिन्हें बुखार आ गया है, जिनको कोई शारीरिक व्याधि है, वो कई बार बीमारी के बढ़ने पर वो कई बार अस्पष्ट बातें करती हैं। उनका जब मानसिक संतुलन बिगड़ जाता है तो ऐसी चीजें बोलती हैं जिस पर लोगों को भरोसा होता है। तो इस पर हमको यकीन करने की जरूरत नहीं है। मैं बताना चाहूंगा कि हम लोग हरेली में, छत्तीसगढ़ में पिछले 30 वर्षों से गांव जाते हैं, क्योंकि यहाँ पर एक मान्यता है कि रात में कोई महिला निकलती है, जादू करती है और गांव में घूमती है और शमशान जाती है। पिछले 25-30 वर्षों से हमको कोई भी ऐसी महिला नहीं मिली जो इस प्रकार की संदिग्ध हो। जबकि ऐसे बहुत सारे लोग मिले, पुरुष मिले, जो लोगों को तंग करते थे, गांव में लूटपाट करते थे। एक और इससे जुड़ी बात है कि आज भी कई स्थानों पर लोग गांव को बांधने की परंपरा करते हैं, कि गांव के चारों ओर कील बांधते हैं और मंत्र तंत्र से बांधते हैं। जबकि हम जब गांव जाते हैं तो हम देखते हैं इतनी गंदगी होती है, वहां पर तालाबों का पानी है, उसी तालाब के पानी को जानवर भी पी रहे हैं और लोग भी पी रहे हैं, बीमार हो रहे हैं। तो उसमें हम इस दौरान अभियान चलाते हैं, हरेली के समय लोगों से बातचीत करते हैं, उनको बताते हैं कि अगर आप सफाई का मंत्र पढ़ेंगे, आप अगर साफ सफाई से रहेंगे, तो आपको कोई भी प्रकार के ताबीज की जरूरत नहीं है, आपको किसी प्रकार के अंधविश्वास को मानने की जरूरत नहीं है। और ऐसा करीब 2000 से अधिक गांव में हमारी सभाएं हुई हैं और हम जाकर के बातचीत करते हैं। और जो भूत प्रेत की जो बात होती है, इसके जो ताबीज देते हैं, ये पूरी तरह से भ्रामक है। यह कई बार, एक बार मनोवैज्ञानिक रूप से अगर आदमी डरता है, अगर वो किसी चीज से भयभीत होता है, तो किसी वो अनजानी शक्ति की कल्पना करता है, भूत की, प्रेत की। और जब बहुत अधिक डरता है तो उसको अलग अलग लक्षण प्रकट होने लगते हैं, शारीरिक लक्षण प्रकट होने लगते हैं, डरने लगता है और उसके बीमारी जैसी चीजें होती हैं। लेकिन अगर किसी अच्छे मनोचिकित्सक को दिखाया जाता है, उसको वापस समाज में अगर सही ढंग से वापस लाना है, तो उसका एक उपचार होने की आवश्यकता है। जो लोग ऐसे लोग अंधविश्वास बढ़ाते हैं, बाबा बन करके, अलग अलग प्रकार के तांत्रिक बन करके, वो उनको ठगने का काम करते हैं। एक अंधविश्वास है बिल्ली के रास्ता काटने की जो बात होती है, जो बचपन से लोग सुनते आ रहे हैं, जो आज भी हमारे पास लोग आते हैं, अशुभ हो जाएगा। कई बार बहुत सारे ऐसे कार के ड्राइवर हैं, वो कार रोक देते हैं कि बिल्ली रास्ता काटेगी साहब, लेकिन ऐसा कुछ नहीं है। यह पूरी तरह से एक भ्रामक बात है। शनिवार को अलग अलग प्रकार की बातें होती हैं, लोहा मत खरीदो, तेल मत खरीदो, इसमें अंधविश्वास कुछ बुरा हो जाएगा। एक अंधविश्वास है कि रात में अगर झाड़ू लगा दी जाए तो घर की संपत्ति समाप्त हो जाएगी, एक अंधविश्वास था। इसका कोई वैज्ञानिक आधार नहीं है। 13 के अंक को अशुभ मानते हैं, ऐसा अलग अलग जो ज्योतिष के लोग बात करते हैं, ये अंक शुभ है, अंक अशुभ है। जितने भी अंक हैं 0 से लेकर 9 तक के सभी अंक जो है मनुष्यों ने बनाए हैं, और यह एक क्रमबद्ध संख्या है। इसमें किसी भी अंक के अशुभ होने की जो बात है पूरी तरह से गलत है। एक सामाजिक अंधविश्वास और है, नमक अगर लोग बोलते हैं हाथ मिलेंगे तो झगड़ा हो जाएगा, यह पूरी तरह से भ्रामक है। क्योंकि नमक का उपयोग आप अपने इलाज के दौरान भी कई चीजों में करते हैं। शरीर में छिपकली गिरने को बहुत सारे लोग अशुभ मानते हैं। ऐसा भी नहीं है, छिपकली भी अगर आप देखते हैं तो उसमें आप सिर्फ अगर नहा लेते हैं तो आपकी जो समस्या है वो दूर हो सकती है, उसमें कोई अशुभ जैसी बात नहीं है। नींबू मिर्च लटका करके नजर लगने की जो मान्यता होती है, कि बहुत सारे स्थानों पर आपने देखा होगा कि बहुत सारे दुकानों पर, वाहनों पर लोग नींबू मिर्च लटकाते हैं और उससे लगता है कि उनको नजर नहीं लगेगी। कई सारे लोग अपने गाड़ियों में, यहाँ हरेली में ऐसा होता है कि लोग अपने अपने वाहनों में नीम की पत्ती लगाते हैं, नींबू मिर्च लगाते हैं कि नजर नहीं लगेगी, जबकि पूरी तरह से भ्रामक है। मैं आपको यह बताना चाहूंगा कि जो नजर की जो बात होती है, इसका कोई वैज्ञानिक आधार नहीं होता है। बहुत सारे लोग शव यात्रा दिखने को शुभ मानते हैं, जबकि अशुभ होता है। समझ लीजिए, इस परिवार के लिए तो अशुभ ही है। चमत्कारिक अंगूठी और खास अंगूठी या ताबीज पहनने पर जो चमत्कारिक सफलता की जो मान्यता होती है, समाचार पत्रों में ऐसे विज्ञापन भरे रहते हैं। गया के पास के बहुत सारे विज्ञापन आते हैं कि आप ऐसे अंगूठी को पहनेंगे तो आपको पूरी सफलताएं मिल जाएंगी। अगर आप इस ताबीज को पहनेंगे तो आपके कोर्ट के मुकदमे जो हैं वो आज आप मनचाही शादी हो जाएगी, मुकदमे जीत जाएंगे, भांति भांति की बातें होती हैं, परीक्षा में सफल हो जाएंगे। इस रुद्राक्ष को बांध लोगे तो आप पास हो जाओगे, पढ़ने की जरूरत नहीं होगी। लेकिन यह सब जो बातें हैं, अगर कोई व्यक्ति अपनी मेहनत करता है तो उसको कोई नहीं रोक सकता। मैं आपको इस संबंध में एक उदाहरण देना चाहूंगा। जब हमारे पूर्व राष्ट्रपति एपीजे कलाम साहब जब अपना नामांकन भरने जा रहे थे, तब उनसे संबंधित मंत्रियों ने कहा था कि आप बताइए आपको कौन से मुहूर्त में नामांकन भरना है, आप अवश्य जीत जाएंगे, हम एक अच्छा मुहूर्त निकालते हैं। तब जो हमारे राष्ट्रपति महोदय जो बने थे कलाम साहब, उन्होंने यह कहा कि जब तक कि पृथ्वी अपने अक्ष पे घूमते रहेगी, दिन और रात होते रहेंगे, मौसम बदलते रहेगा, तब तक मेरे लिए हर मौसम हर दिन शुभ है, मैं कभी भी नामांकन भर सकता हूँ। और हम सबको मालूम है कि वे देश के सबसे लोकप्रिय राष्ट्रपतियों में से एक थे और उन्होंने देश में सुधार के बहुत सारे काम किए हैं। चमत्कारिक सिक्के से जो खजाना प्राप्ति की जो बात होती है, यह इसमें बहुत सारे लोग हैं आज भी ज्वेलरों के यहाँ घूमते रहते हैं कि हमको 2020 1919 2121 के सिक्के चाहिए ताकि हमको खजाना मिल सके, लेकिन ये पूरी तरह से भ्रामक है। एक अंधविश्वास है सवेरे सवेरे चेहरा देखने पर अशुभ होने की जो भ्रम होता है। बहुत सारे लोग हैं ऐसी बातें करते हैं कि अगर मैं सवेरे घर से बाहर जा रहा था कि कोई व्यक्ति दिख जाता है तो मेरा दिन खराब हो जाता है। ये एक मनोवैज्ञानिक समस्या है। एक मानसिक रूप से जो लोग परेशान होते हैं वो ऐसी बातें करते हैं, क्योंकि हम यह जान लें कि कोई भी व्यक्ति जो है जब भी सवेरे उठता है तो सबसे पहले वो घर में अपना चेहरा देखता है, अपनी पत्नी का देखता है, अपने बच्चों का देखता है, लेकिन वो कभी किसी को खराब नहीं कहता है। वो टारगेट करता है एक ऐसे व्यक्ति को जो घर के बाहर एक गरीब व्यक्ति है, जो विकलांग है, जो कुरूप है। वो अगर उससे कुछ कह देता है तो उसको लगता है कि मेरा दिन खराब हो गया। पूरी तरह से अंधविश्वास है। नजर लगने से बचने के लिए जो काला टीका लगाने की बात होती है, हमारे पास ऐसे बहुत सारे मामले आते हैं जिसमें लोग कहते हैं कि भाई हमने जो काला टीका लगाया इससे नजर नहीं लगेगी। यह पूरी तरह से भ्रामक है। मैं इसमें आपको आगे और बातचीत करूंगा। लकड़ी को टच वुड वाली जो बात होती है कि नाउ मी दी नॉट टच वुड, टच वुड जो खासकर चीजें हैं ये युवाओं के बीच लोकप्रिय है, लेकिन क्या है कि इस इतिहासकारों को इनके बारे में बहुत अधिक पता नहीं है। लेकिन यह है कि इस धारणा का जो जन्म है कि जो लोग क्रूज पे हाथ पर रख करके बात करते उससे वो होगा। घर के बाहर घोड़े की नाल टांगने की जो बात होती है, बहुत सारे स्थानों पर आज देखिए लोग इतने महंगे घर बनाते हैं उसके बाद भी वो एक घोड़े की नाल टांग देते हैं। क्यों टांग देते हैं? कि उनके घर में अशुभ ना हो, अशांति ना हो। तो ये एक पुराना अंधविश्वास है, क्योंकि एक इस पे एक थ्योरी है कि मध्यकाल में लोग ऐसा मानते थे कि जो कुछ भूत प्रेत हैं वो घोड़े की आवाज से डरते हैं, इसलिए उनके आगमन को रोकने के लिए वो घोड़े के पैर की नाल को लगाते थे। लेकिन इस परंपरा के कारण आज बहुत सारे जो घोड़े हैं राजस्थान में हैं, उनके खुर में जो नाले रहती है वो हर 2-3 दिन बदल दी जाती है। लोग उस नालों को ले जाते हैं और इससे घोड़ों के खुर में घाव होते हैं, उनको परेशानी होती है। गिरते हुए तारे को देख करके कुछ मांगना, कुछ फिल्मों ने इस चीज को बहुत प्रचलित किया है कि अगर कोई तारा टूटता है तो उससे अगर मांगा जाता है उसकी मान्यता पूरी होती है। ये बहुत पुराना अंधविश्वास है। ये पहली शताब्दी से जुड़ा है, क्योंकि उस शताब्दी के लोग ये मानकर चलते थे कि आसमान में अगर कोई तारा टूटता है तो उस तारे के साथ में देवता जो है जमीन पर उतरते हैं, उससे जो मांगो तो हो जाता है। लेकिन आज हमको सब मालूम है कि यह इस खगोल शास्त्र में हमने पढ़ा है कि इस दुनिया में है, इस संसार में है, कितने तारे प्रतिवर्ष और टूटते हैं, बनते हैं, नए नए तारे जन्म लेते हैं और वो धीरे धीरे गिरते हैं। इसमें कोई चमत्कार ऐसी बात नहीं है। सांप से जुड़े बहुत सारे अंधविश्वास हैं कि सांप जो है वो बीन से नाचता है, सांप बदला लेता है, सांप इच्छाधारी सांप होता है, सांप अपने रूप बदल लेता है। और इस घटना के कारण बहुत सारे लोगों ने अपनी जानें भी खोई हैं। मैं आपको बताना चाहूंगा कि सांप के बिहार में एक घटना हुई थी, इसमें एक महिला को इसलिए मार दिया गया कि वो उसको कहते थे कि वो सांप बन करके जाती थी और लोगों को डसती थी। की घटना है, जबकि वास्तव में ऐसा कुछ नहीं है। किसी भी व्यक्ति में ऐसी ताकत नहीं होती है कि वो अपना स्वरूप इस प्रकार बदल सके। कोई ना कोई व्यक्ति इंसान से जानवर बन सकता है, ना जानवर से पक्षी बन सकता है, कोई कुछ नहीं कर सकता है। जिस हम उसमें हैं उसी स्वरूप में आजीवन रहते हैं। जो तथाकथित चमत्कार होते हैं, ग्रामीण अंचल में आज भी ऐसे बहुत सारे मामले आते हैं जो लोग कुछ चमत्कार दिखा करके लोगों को ठगते हैं। और कोई पर्ची निकालता है तो कोई अलग अलग प्रकार के ताबीज की बात करता है, कोई ट्रिक्स दिखाता है। इसके बारे में हम लोगों को जागरूक करते हैं, हम उनको बताते हैं कि बहुत सारे जो चमत्कार होते हैं वो हाथ की सफाई से होते हैं, बहुत सारे चमत्कार होते हैं वो आपके मानसिक चीजों को पढ़ करके होते हैं। आपने देखा होगा मेंटलिस्ट लोगों ने अभी कुछ दिनों पहले करके बताया है कि वो कैसे मन की बातें जान जाते हैं। बहुत सारे चमत्कार जो होते हैं केमिकल रिएक्शन से होते हैं, हम इसके लिए साइंस कॉलेज से और अलग अलग जगहों से हम केमिकल्स लेते हैं, जैसे नींबू से खून निकलने की बात होती है या दृश्य शाही जैसी बात होती है। हम लोगों को करके दिखाते हैं कि इसमें आप चमत्कार पे मत पड़िए, यह एक प्रकार की रासायनिक प्रक्रिया है और जिस व्यक्ति को उस रासायनिक प्रक्रिया के बारे में जानकारी नहीं होती उसके लिए ये चमत्कार है, लेकिन जिस व्यक्ति को जानकारी है उसके लिए सामान्य सी घटना है। जैसे हमारे लिए, मैं नेत्र विशेषज्ञ हूँ, हमारे लिए आंख का ऑपरेशन करना सामान्य है और व्यक्ति को दूसरे दिन दिखने लगता है। अगर कोई व्यक्ति मोबाइल बनाता है तो उसके लिए मोबाइल बनाना सामान्य सी चीज है, कोई व्यक्ति अगर टीवी बनाता है उसके लिए टीवी बनाना सामान्य चीज है, लेकिन दूसरे व्यक्ति के लिए वो चमत्कार है। क्योंकि हम जब गाँव में जाते हैं तो हम ये चीज बात भी करते हैं कि बहुत सारे लोग हैं जो देखिए आप वहां पर ताबीज बांधे दिखते हैं, कोई लहसुन बांध कर दिखता है तो कोई अलग अलग प्रकार के काला धागा बांधता है। तो हम उनसे ये बातचीत भी करते हैं कि भाई आपके घर में जो सामान है उस सामान के लिए आप, अगर सामान खराब होता है तो आप क्या करते हैं, तब बताते हैं कि भाई हमारा टीवी खराब होता है तो हम टीवी के मैकेनिक के पास लेके जाते हैं, अगर हमारा ट्यूबलाइट खराब होती है तो हम ट्यूबलाइट के मैकेनिक को दिखाते हैं, अगर हमारा स्पीकर खराब होता है तो हम स्पीकर को उसके मैकेनिक के पास जाते हैं, पंचर अगर गाड़ी होती है तो हम पंचर के मैकेनिक के पास जाते हैं। तो हर चीज के लिए अलग व्यक्ति को चुनना चाहते हैं। लेकिन अगर कोई व्यक्ति बीमार होता है तो यह जो हमारा जो शरीर है यह भी एक प्रकार की मशीन है, इस मशीन के अलग अलग अंग हैं। अगर इस शरीर का कोई भी अंग खराब होता है, कोई भी मशीनरी खराब होती है तो हमको उसके डॉक्टर के पास जाना चाहिए और उससे हमको इलाज कराना चाहिए। एक ही प्रकार का ताबीज, एक ही प्रकार की झाड़ फूक, एक ही प्रकार का नींबू मिर्च, ये चीजें हैं वो किसी व्यक्ति को कैसे ठीक कर सकती हैं, ये लोगों को समझाने की आवश्यकता है। और स्वास्थ्य से जुड़े हुए जो अंधविश्वास हैं, मैं बताना चाहूंगा कि समाज से जो बहुत सारे अंधविश्वास हैं। जैसे कोरोना के समय देखा गया कि बहुत सारे लोगों ने अलग अलग प्रकार की चिकित्सा भी प्रयोग की, किसी ने झाड़ फूक भी की, किसी ने कोरोना देवी का मंदिर बनाया, किसी ने कोरोना के लिए गंगा नदी के किनारे पूजा भी की, अलग अलग प्रकार के लोगों ने ताबीज भी बनाए। लेकिन कोरोना जो ठीक हुआ वो जब लोगों ने आपसी संपर्क कम किया, मिलना जुलना कम किया, जब उसमें कर्फ्यू लगा उस समय कम हुआ, जब वैक्सीन आई तो कोरोना ठीक हुआ, वो किसी झाड़ फूक से कम नहीं हुआ। जो पीलिया की झाड़ फूक होती है, जो पीलिया एक ऐसी बीमारी है जो हमारे लीवर में खराबी से होती है, लेकिन आज भी बहुत सारे लोग हैं जो पीलिया को झाड़ फूक करके ठीक करने की बात करते हैं। तो पीलिया के संबंध में मैं बताना चाहूंगा कि जो पीलिया एक ऐसी बीमारी है जिसमें हमारा लीवर जो है वो कमजोर हो जाता है, वह क्षतिग्रस्त हो जाता है, और वह जो होता है वो कई बार छोटे बच्चों में होता है, उनमें जब उनके हीमोग्लोबिन कम होने लगता है तब उनमें होता है। कोई व्यक्ति अगर बहुत शराब का सेवन करता है तो उसका कम होता है, और संक्रमण से हमारा जो लीवर है वो लीवर खराब होता है। तो इसके कारण जो लीवर खराब होता है उसके कारण शरीर में पीलापन आता है, पेशाब पीली होने लगती है, शरीर का रंग पीला होता है। इसको किसी भी प्रकार से धो करके, पानी से धो करके, तेल से धो करके इसको ठीक नहीं किया जा सकता। तो जो लोग भी कोरोना की झाड़ फूक की बात करते हैं, झाड़ फूक करते हैं, तो पूरी तरह से वो अवैज्ञानिक है। जो कुष्ठ रोग की बात है, जब कुष्ठ रोग के बारे में जानकारी नहीं थी तब बहुत सारे स्थानों पर हमने देखा है देवभोग के पास में कि लोगों को गांव के बाहर छोड़ देते, इसको किसी देवी श्राप से इसके हाथ पैर गल गए। लेकिन जब बाद में पता चला कि माइकोबैक्टीरियम लेप्री नाम का इसके लिए एक जो ऑर्गेनिज्म है वो इससे फैलता है, तब लोगों ने लेप्रोसी का इलाज करना शुरू हुआ। आज बहुत सारे लोग हैं जिनको इस अवस्था में नहीं पहुंचते कि उनके हाथ पैर गल जाएं, उनके हाथ पैर में पूरी तरह से नुकसान हो जाए, और वो समाज के काम आ जाते हैं। एक अंधविश्वास है, मैं कहना चाहूंगा बहुत सारे लोग इस बात को मानते हैं, काजल लगाने से आंखों के बड़ी होने की जो बात होती है और नजर तेज होने का जो भरोसा होता है, यह भी एक प्रकार से गैर साइंटिफिक बात होती है। काजल लगाने से आपकी आंखें बड़ी नहीं होती, काजल लगाने से आपकी आंखें बड़ी लगती हैं, और काजल लगाने से किसी व्यक्ति की नजर भी तेज नहीं होगी, क्योंकि जो काजल है एक प्रकार का कॉस्मेटिक है। अगर किसी व्यक्ति को अपनी या अपने बच्चों की नजर को अच्छी रखना है तो आवश्यक है कि वह अच्छे पोषण ले, अच्छे पौष्टिक आहार का सेवन करे, उसके खाने में हरी सब्जियां हो, फल हो। बहुत जरूरी नहीं कि हमको महंगी चीजों को लेना है, सस्ती चीजों से भी हमारा काम चल सकता है, लेकिन विटामिन ए होना चाहिए, हमारे आसपास की चीजों में भरपूर रूप से मिलता है, और उसका सेवन करें। तो जो आजकल के बच्चे हैं लगातार मोबाइल पर बैठते हैं, कंप्यूटर पर बैठते हैं, उससे भी मायोपिया बढ़ता है। जो बहुत छोटे बच्चों को जो मोबाइल उनकी माताएं पकड़ाती हैं, और उसका जो स्क्रीन रंगीन होता है वो भी हमारी आंखों के मायोपिया को बढ़ाता है। जो गर्म हंसिया या सलाख से जो आंका जाता है उससे ठीक हो जाएगी बात होती है। अभी शहडोल में 2 दिनों पहले की बात है, वहाँ 2 बच्चों की मृत्यु हुई है। वहाँ निमोनिया होने पर वहां पर बैगाओं ने उन्हें सलाख से दागा था। ऐसी घटना छत्तीसगढ़ के आसपास में और बहुत सारे स्थानों पर होती है, चूड़ी से दाग दिया जाता है। जबकि इस प्रकार के दागने से कोई बीमारी ठीक नहीं होती है, कोई उपचार नहीं है, यह पूरी तरह से अंधविश्वास है। सर्दी होने पर या सफेद दाग होने पर बहुत सारे लोग जो है अलग अलग प्रकार की माला, काला धागा, ताबीज पहनते हैं, ये पूरी तरह से गलत है। मानसिक रोगी जो होते हैं उनको लोग भूत प्रेत से संबंधित मानते हैं और देवी संचार के जो मामले होते हैं कि किसी कई बार ऐसे लोग आपके आसपास भी ऐसे लोग होते हैं क्योंकि आकाश को देवी आती है, ऐसा आता है, लेकिन वास्तव में यह एक प्रकार का मनोवैज्ञानिक मामला होता है, इसका उपचार होना चाहिए। कुत्ते काटने की जो बातचीत होती है, कुत्ते काटने की जो बातचीत होती है, सर्पदंश की झाड़ फूक होती है, यह भी पूरी तरह से एक प्रकार का अंधविश्वास होता है। क्योंकि जब चिकित्सा प्रणाली विकसित नहीं हुई थी और लोगों के पास कोई साधन नहीं था, तब लोग जानवरों से दंश होने पर वे इलाज करते थे। लेकिन अब हमारे पास में जो वैक्सीन है, उस वैक्सीन का उपयोग करना चाहिए ताकि लोग ठीक ढंग से रह सकें। और छोटे बच्चों के बीमार होने पर जो लोग बात करते हैं कि किसी ने नजर लगा दिया होगा, जादू टोना कर दिया होगा, ये पूरी तरह से अंधविश्वास है। मैं नेत्र चिकित्सक हूं, मैं आपसे बताना चाहूंगा कि जो नजर होती है वो 2 प्रकार की होती है। एक नजर जो अच्छी नजर जिसमें हमको बिना चश्मे के दिखता है, पूरी तरह से आप स्पष्ट देख लेते हैं। दूसरी नजर जो होती है कमजोर नजर होती है, जिस नजर में आपको चश्मा लगा करके साफ दिखता है, चश्मा नहीं है से अगर नहीं हो रहा है तो आपका ऑपरेशन होता है, आपको दवाई दी जाती है, उससे साफ दिखता है। तो लेकिन ऐसी कोई नजर नहीं होती जिससे किसी व्यक्ति को कोई व्यक्ति दूसरा व्यक्ति बीमार कर सके। सिर्फ इसी नजर लगने के कारण देश में बहुत सारे आपराधिक मामले घटते हैं और उसका कोई फायदा नहीं होता है। जो मीजल्स की बात होती है, मीजल्स है, माता निकलने की जो बात होती है, इस पे लोग कई बार इलाज नहीं कराते हैं, कहते हैं इसकी शांति के लिए हम करेंगे, अनुष्ठान करेंगे, लेकिन यह भी एक प्रकार से भ्रामक बात है। जो मीजल्स है, जो स्मॉल पॉक्स था, चिकन पॉक्स है, ये एक प्रकार के वायरल संक्रमण है और ये एक कुछ समय के बाद स्वतः ठीक होने लगते हैं। ये ध्यान रखने की आवश्यकता होती है कि अगर व्यक्ति कोई बीमार है तो उसको और कॉम्प्लिकेशन ना हो, इसलिए उसको एक सिम्टोमेटिक इलाज दिया जाता है, उसको किसी प्रकार की झाड़ फूक की जरूरत नहीं है। संतान न होने पर जो छोटे बच्चों की जो बलि की मान्यता होती है, बहुत सारे मामले आपने देखा होगा बलि प्रथा की। इटारसी से कुछ दिनों पहले एक मामला आया था मध्य प्रदेश से, कई बार हरियाणा से ऐसे मामले आते हैं जिसमें कोई बैगा के कहने पर लोग बलि देते हैं छोटे बच्चों के, किसों के घर बच्चे हो जाएंगे, लेकिन यह पूरी तरह से अंधविश्वास जैसी बात है। किसी अगर आप दूसरे व्यक्ति की जान लेते हैं तो निश्चित रूप से आप जेल जाने वाले हैं, आपके घर में बच्चे अगर कुछ नहीं होने वाले हैं। मिर्गी के जो मरीज होते हैं उनको जूते सुंघा के जो ठीक करने का एक अंधविश्वास है, ये अभी भी बहुत सारे स्थानों पर चलता है, ग्रामीण अंचल में देखने में आता है। मिर्गी जो है, मैं आपको बताना चाहूंगा कि जो मिर्गी की जो बीमारी होती है उसमें एक मिर्गी ऐसी होती है जिसमें आदमी कुछ सेकंड में होश में आता है, गिरता है, उसको झटके आते होश में आता है। तो उसमें आप उसमें किसी प्रकार का इलाज नहीं भी करते तो भी ठीक हो जाता है। तो उसमें कई बार लोग उसको जूते सुंघाते हैं, गाँव में उसको अलग अलग प्रकार से इलाज करने की कोशिश करते हैं। मिर्गी की जो दूसरी प्रकार की बीमारी होती है, मेजर होता है, तीसरा स्टेटस एपिलेप्टिकस होता है, उसमें व्यक्ति को अस्पताल में भर्ती करना चाहिए। क्योंकि अगर व्यक्ति अर्धमूर्छित है, जमीन पर गिरा हुआ है, तो वह व्यक्ति अपनी जीभ काट सकता है, उसकी सांस रुक सकती है, इसलिए उसको हमको अस्पताल में पहुंचाना चाहिए। कई बार मिर्गी के धोखे में लोग ऐसे व्यक्ति को भी जूते सुंघाते हैं जिनको हार्ट अटैक आया है, उनको पैरालिसिस हुआ है, सड़क पर गिर गया है। इस प्रकार से हमको चीजों से बचने की आवश्यकता होती है। जो दमे की बात होती है, इसमें बहुत सारे अंधविश्वास चिकित्सा से जुड़े हुए हैं। जो दमे का जो इलाज होता है, दमे के इलाज के लिए जो मछली का विज्ञापन आता है कि मछली खाएंगे तो आप ठीक हो जाएंगे, ऐसे कुछ ठगों को हमने रायपुर में पकड़ा भी है। और ऐसे बहुत सारे लोग जो है मछलियों को खिलाते जरूर हैं, लेकिन मछलियों के साथ में वो मछलियों के मुंह पर वो डेकाड्रान में डुबा के रखते हैं, उनको एस्थिलीन में डुबा के रखते हैं, डेयरी फार्म में डुबा के रखते हैं मछलियों को, जिससे व्यक्ति को कुछ समय के लिए अच्छा लगता है, लेकिन वह व्यक्ति किसी के गले को नहीं साफ कर सकती है मछली, वो मछली जो है पेट में जाती है। लोग जो ऐसी बातें करते हैं कि मछली से आपका गला साफ होगा, फेफड़े साफ हो जाएंगे, सांस की नली साफ हो जाएगी, भ्रामक है। अगर हम सब विज्ञान के विद्यार्थियों को यह बात मालूम है कि अगर कोई व्यक्ति धोखे से भी अगर एक छोटा सा चना या मूंगफली भी अगर उसके सांस नली में चले जाता है तो उस व्यक्ति की मृत्यु हो सकती है, तो मछली तो उसकी सांस नली में जो जाने की बात है वो पूरी तरह से गलत है। गर्भवती महिला को नजर लगने से बचाने के लिए अंधेरे कमरे में जो रखने की जो बात होती है, ऐसा राजस्थान में बहुत सारे मामले में होता है कि महिला को बुरी नजर न लगे इसलिए उनको गर्भावस्था के समय में अंधेरे कमरे में रखा जाता है, उनको खाने पीने की चीजों, उनको वहां पर विटामिन डी की कमी हो जाती है, वो मानसिक रूप से परेशान हो जाती हैं, यह पूरी तरह अंधविश्वास है। ऐसी कोई नजर नहीं होती जिसमें उसको बचाने के लिए अंधेरे कमरे में रखने की जरूरत है। उसको खुली हवा दीजिए, उसको अच्छा खाना दीजिए, उसको पौष्टिक आहार दीजिए। एक अंधविश्वास देखिए बहुत सारे छत्तीसगढ़ और उड़ीसा के प्रदेशों में देखने में आता है, बिजली गिरने पर गोबर का लेप लगाने की जो बात होती है। बहुत से लोग मानकर चलते हैं कि बिजली अगर गिरती है तो उस व्यक्ति को शरीर गर्म हो जाता है, उसको गोबर का लेप लगाते हैं और ठीक हो जाएगा। लेकिन वास्तव में बिजली जो गिरती है उसमें हजारों वाट का करंट होता है और किसी व्यक्ति को उससे हार्ट फेल या उसको ब्रेन स्ट्रोक हो सकता है। उसको जितनी जल्दी हो सके उसको अस्पताल पहुंचाना चाहिए। अगर उसको किसी गांव के स्थान पर हम गोबर के गड्ढे पर ले आते हैं, गोबर उसके ऊपर लेप लगाते हैं कि ठीक हो जाएगा, तो पूरी तरह से यह गलत है और भ्रामक है। अभी कुछ समय अभी देखा है कि बहुत सारे लोग ऐसी भी बातें हैं कि लोग सीजर का मुहूर्त भी निकालने लगे हैं कि सीजर करवाना है, मुहूर्त निकाल के करेंगे। लेकिन क्या सीजर जो किए जाते हैं वो व्यक्ति की आवश्यकता के हिसाब से किए जाते हैं कि अगर बच्चे की ठीक नॉर्मल डिलीवरी नहीं हो रही है तो उसका सीजर किया जाए। तो यकीन करते हैं, तो गाँव में तो खैर साधन नहीं है। शहरों में कई बार ऐसा भी होता है कि परंपराओं को मानने का जो दबाव होता है और किसी अनजानी ताकत से अहित होने का जो भय होता है कि कुछ हो जाएगा, इससे कई बार लोग अंधविश्वास पे भरोसा करते हैं। स्वास्थ्य लाभ की उम्मीद, कुछ बीमारियां ऐसी होती हैं हमारे बीच में जो तुरंत ठीक नहीं होती, उस पर लंबे समय तक हमको लगातार उपचार लेना पड़ता है। जैसे थायराइड, उसमें दवा खाना पड़ता है, ब्लड प्रेशर में दवा खाना पड़ता है, शुगर में, डायबिटीज में दवा खाना पड़ता है। लेकिन बहुत सारे लोग चाहते हैं कि वो ऐसी कोई उनको ऐसी चीज मिल जाए, जादुई चीज, जिससे उनका ब्लड प्रेशर ठीक हो जाए। इसलिए लोग कई बार अंधविश्वास पे यकीन करते हैं और ऐसे कई बार काले धागे, ताबीज आदि बांधकर घूमते हैं। आर्थिक लाभ की उम्मीद, बहुत सारे पढ़े लिखे लोग हैं वो भी लॉटरी के चक्कर में फंसते हैं, ऐसी बातें अनुष्ठान करना चाहते हैं कि उनसे उनको तुरंत सफलता मिल जाएगी, तुरंत प्रमोशन हो जाएगा। लेकिन जो व्यक्ति सही ढंग से मेहनत करता है वो निश्चित रूप से धीरे धीरे सफल होता है। चमत्कारी सफलता की जो बात होती है, एक दिन में आपका हो जाएगा, ऐसा हो जाएगा, लेकिन जो बातें कहानी के रूप में ठीक लगती है, लेकिन वास्तव में आप जो लोग सही ढंग से मेहनत करते हैं वो एक सही स्थान पर पहुंचते हैं। अंधविश्वासों को भाव कैसे देता है करते हैं जब हमारे घर में जन्मता है मामला, शरीर बहुत है, कॉम्पिटिशन बहुत ज्यादा है, सीटें बहुत कम होती है, जो अक्षर की की बात करते हैं,
मुझे लगता है कुछ कनेक्टिविटी प्रॉब्लम आ रही है सर के साथ।
मैम उनको फोन करके बोलिए ना, फिर से जुड़ेंगी, वो लीव हो गए। रेणु महेश्वरी मैडम, हाँ ज्वाइन किए सर। सर प्लीज अनम्यूट योरसेल्फ।
मैं उनको फोन करती हूँ, पर फिर मुझको फिर से मीटिंग में जुड़ना होगा क्योंकि मैं फोन से ही जुड़ी हूँ।
ओके,
नहीं मैम, वो ज्वाइन कर लिए।
अनम्यूट कर लीजिए सर।
हाँ, ठीक है। सर आप अपने आप को अनम्यूट कर लीजिए। मिश्रा सर।
हाँ हाँ
हाँ हाँ, अब आपकी आवाज आई। जी जी जी।
अभी ठीक है।
हाँ आप, हाँ अभी वीडियो नहीं दिख रहा है आपका, आवाज आ रही है।
अच्छा,
हाँ, वीडियो थोड़ा देख लीजिए, वीडियो स्टार्ट कर लीजिए। हाँ ठीक है सर, ओके।
नहीं आ रहा है।
आवाज नहीं आ रही सर आपकी, फिर से।
अनम्यूट करिए, अनम्यूट करिए।
अभी आज
जी
हाँ
जी
अब आ रही, जी जी।
जो समाज का प्रभावशाली वर्ग है कई बार वो अंधविश्वासों का अनुसरण करता है और सार्वजनिक रूप से उनकी वकालत करता है। जैसे बहुत सारे समय ऐसा भी होता है कि लोग जो है बड़े बड़े अस्पतालों में जाकर ठीक होते हैं, लेकिन वो प्रचार प्रसार किसी ऐसे तीर्थ स्थल का करते हैं कि
सर आई थिंक देयर इज अ नेटवर्क प्रॉब्लम, आपकी आवाज नहीं आ रही क्लियर। हेलो, एक्सक्यूज मी सर, सर आपकी आवाज क्लियर नहीं है।
सर आपकी आवाज क्लियर नहीं है। सर आपकी आवाज क्लियर नहीं है।
भ्रम आता है। जो बहुत सारी फिल्में लोगों को बढ़ावा देते हैं, कई सारे जो टीवी में सीरियल आते हैं जो अंधविश्वास का
सर फिर से लेफ्ट हो गए, शायद ज्वाइन करने की कोशिश कर रहे हैं।
एंड बैंडविड्थ इशू दिख रहा है।
कोई डिस्टरबेंस होगा शायद उधर, कनेक्टिविटी प्रॉब्लम हो सकता है। सर आपकी आवाज नहीं आ रही है।
सर आउट हो गए हैं।
अभी लेफ्ट है मैम, सर अभी नहीं।
ओके, बात करके देखूं क्या करूं।
एंड पे
एक बार
नेटवर्क इशू
बात-
काफी समय से आ रहा है मैम।
दीपाली, if you have phone, then call sir.
आई डोंट हैव नंबर मैम।
दी नंबर भेजिए, सेंड कीजिए।
मेरे को मीटिंग से लीव होना पड़ेगा।
No, don't leave the
विदाउट
meeting.
लीविंग कर सकते हैं मैम।
Don't leave the
देख
meeting.
कोशिश करती हूँ, कोशिश करती हूँ।
हाँ सर ज्वाइन किया है
हाँ, नंबर लिखना है।
मैम, सर ने ज्वाइन किया है मैम।
ओके,
हेलो,
हाँ सर।
उसको
हाँ अभी ठीक, अभी जुड़े हैं आप।
अभी आ रही है आवाज मेरी?
हाँ आ रही है, पर थोड़ा डिस्टरबेंस है।
अच्छा अच्छा,
हाँ अभी ठीक है, अभी ठीक है
ठीक है।
जी।
अंधविश्वासों को जब हम हटाने की बात करते हैं तो अंधविश्वासों को कैसे खत्म किया सकते हैं। शिक्षा का प्रचार प्रसार जो है, स्वास्थ्य के प्रति जागरूकता बढ़ाना, वैज्ञानिक दृष्टिकोण की जो हम बात करते हैं, किसी भी घटना विशेष की जो जांच पड़ताल होती है, हम इस बारे में भी चर्चा करते हैं। कई बार हम जाते हैं गाँव में, कई बार ऐसी चीजों को जो है प्राकृतिक घटनाओं को लोग चमत्कार के रूप में बताते हैं। जैसे कुछ दिनों पहले छत्तीसगढ़ में मामला आया था कि एक पेड़ से जो पानी निकला, उसको लोगों ने कहा कि दूध निकल रहा है, जबकि वास्तव में उसका जो प्रोटोप्लाज्म था, उसके अंदर का जो जीव द्रव्य रहता है, वो संक्रमित हो गया था, उसके कारण वो दूधिया हो गया था। लेकिन गाँव में कुछ लोगों ने कहा कि यहाँ से पेड़ से दूध निकल रहा है और उस दूध को पीने से लोग ठीक हो जाएंगे, जबकि बात पूरी तरह से गलत थी। कथित चमत्कारों का जो प्रदर्शन है, इसके उपचार की जो प्रेरणा होती है, हमारे भारत सरकार का एक संविधान में एक कानून है, रेमेडी एक्ट 1954। इस अधिनियम का उपयोग करके हम लोगों के बीच में से जो सामाजिक अंधविश्वास दूर करने का कोशिश कर सकते हैं। छत्तीसगढ़ राज्य टोनी प्रताड़ना अधिनियम, ऐसा अधिनियम देश के कुछ प्रदेशों में भी बना है, जैसे झारखंड में है, बिहार में है, कि अगर किसी महिला को किसी भी इस प्रकार दंडित किया जा सकता है। मैं यह भी बातचीत करना चाहूंगा कि वैज्ञानिक दृष्टिकोण का अगर विकास होता है तो उससे क्या क्या फायदे हो सकते हैं, जो सामाजिक जागरूकता में वृद्धि होगी। अभी देखिए बहुत सारे लोग हैं वो कई बार एक ही अफवाह की तरफ चले जाते हैं, उसमें कमी होगी, अंधविश्वास और कुरीतियां कम होंगी, जो आत्मनिर्भरता बढ़ेगी, जो भाग्य भरोसे बैठने की जो बात होती है वो कम होगी, स्वास्थ्य चेतना में वृद्धि होगी। जो बहुत सारी छोटी छोटी बीमारियों के मामले में जो ऐसी घटनाएं होती हैं, किसी को बोलने की क्या बीमार हो गया, किसी ने कुछ कर दिया है, किसी को दागा जा रहा है, किसी को गोबर में लपेटा जा रहा है, इस प्रकार की घटनाएं कम होंगी। जो निर्दोष महिलाएं हैं और बच्चों की प्रताड़ना में कमी आएगी। इसमें मेरा यह कहना है कि जो हमारी जो वैज्ञानिक दृष्टिकोण है, ये ऐसी प्रवृत्ति है जिसका मूल आधार किसी घटना की जो पृष्ठभूमि में उपस्थित कार्य को जाना चाहिए। हमारा जो संविधान है उसके अनुच्छेद 51A के अंतर्गत जो है मौलिक कर्तव्यों में से वैज्ञानिक दृष्टिकोण का विकास करना, ये हमारे मौलिक संविधान का अंग है। इसलिए हम में से प्रत्येक नागरिक का यह कर्तव्य है कि वैज्ञानिक दृष्टिकोण के विकास के लिए प्रयास करें। जो हमारे संविधान के निर्माता हैं उन्होंने यही सोचकर वैज्ञानिक दृष्टिकोण को मौलिक कर्तव्यों की सूची में शामिल किया है कि भविष्य में वैज्ञानिक सूचना और ज्ञान में वृद्धि से वैज्ञानिक दृष्टिकोण युक्त चेतना और संपन्न समाज का निर्माण होगा। परंतु वर्तमान में सत्य इससे परे है। इसके लिए हम सबको प्रयास करने की जरूरत है। जब हम अपने क्षेत्र में विज्ञान की आराधना करने वाले वैज्ञानिकों का जो प्रत्यक्ष सामाजिक व्यवहार है वो वैज्ञानिक दृष्टिकोण से ही और लोग प्रेरित होंगे, और जो हमारे बीच में जो युवा वर्ग है, जो हमारे बीच में बच्चे हैं, वो हमारे जो कार्यकलाप है, जो हमारी प्रवृत्ति उस पर भी आधारित होंगे। वैज्ञानिक दृष्टिकोण जो है ये तर्कशीलता से संबंधित रखती है, और वही बात हमको देखना चाहिए जो परिणाम से, प्रयोग से सिद्ध हो सके और जिसमें संबंध स्थापित किया सके। क्योंकि हम देखिए, हम जो विज्ञान में नियम पढ़ते हैं, हम उसको लैब में जाकर के बाकायदा प्रयोग कर पहुंचते हैं। किसी भी अनजानी घटना को, किसी भी अफवाह को, किसी भी व्हाट्सएप के माध्यम में फैलती हुई भ्रामक प्रचार को सही मान करके उसको आगे बढ़ाना, उसको प्रचारित करना पूरी तरह से गलत है, और इससे लोगों का बहुत मौलिक नुकसान होता है। गौरतलब है कि निष्पक्षता, मानवता, लोकतंत्र, समानता और स्वतंत्रता के निर्माण में भी वैज्ञानिक दृष्टिकोण जो है अधिक कारगर सिद्ध होता है। क्योंकि जो ये और आज बड़ी बड़ी अट्टालिकाएं हैं, लोगों के पास बहुत सारे साधन हैं, तो हमको साधनों का उपयोग कर जब हम करते हैं, जब हम अपने जीवन में वैज्ञानिक उपलब्धियों का पूरी तरह से उपयोग करते हैं, तो हमको अपनी सोच भी वैज्ञानिक करने की जरूरत है, वैज्ञानिक दृष्टिकोण को बढ़ाने की जरूरत है। थिंक साइंटिफिकली, एक्ट साइंटिफिकली, उसको सोचने की जरूरत है और हमको वैज्ञानिक ढंग से रखने की जरूरत है। वैज्ञानिक रूप से सोचें, वैज्ञानिक रूप से काम करें और वैज्ञानिक रूप से जिए, ताकि जो पूरी मानवता है उसको एक सफल आयाम मिले। धन्यवाद।
Thank you so much, sir, for the eye-opening talk. You talked about the relevance of scientific temperament in present society, which is dealing with information overload and superstitions. Without thinking scientifically and logically, if we put every information to practice, it will be extremely detrimental for the society. We extend our best wishes to you, sir, for the good work you are doing to curb superstition. I request Renu Maheshwari ma'am to extend vote of thanks to sir.
दीपाली, I would
जी, ma'am.
like to ask sir question.
Okay. Yes.
सर, आपने इतने सारे मतलब ग्राउंड लेवल पे जाके काम किया है आपने, तो शुरुआत में तो बहुत परेशानी हुई होगी। तो हम लोग वो इंस्टेंसेस जानना चाहते हैं जहां पर आपने बहुत मुश्किलों से लोगों को कन्विंस करने की कोशिश की, क्योंकि इतना आसान नहीं होता है। धीरे धीरे चीजें आसान होती है, लेकिन जब शुरुआत में जो आपने स्टेप लिया, पहला हिम्मत दिखाई, उसके बारे में एक आध दो अगर वाक्य आप बता सकें तो सबके लिए इंस्पायरिंग होगा वो।
नहीं, उसमें हमने शुरुआत जब की तो छत्तीसगढ़ में 1995 में हमने काम शुरू किया। यहाँ पर एक पहली घटना हुई, इसमें एक महिला को जादू टोने के आरोप में उसको घर से निकाल के मारा गया था और उसको गले में पत्थर बांध के उसको कुएं में डाल दिया गया था। जब ये घटना सामने आई तब उस समय मैं प्रैक्टिस में नया आया, तो हमने शुरू किया कि यहाँ पर एक संस्था बनाई जाए जो कि लोगों को वैज्ञानिक रूप से जागृत करने का काम करे। तब उस गाँव में हम गए, वहां पर जाकर के लोगों से मिले, लेकिन पहले वहां पर कोई कुछ बताने को तैयार नहीं था। फिर धीरे से कुछ लोगों को हमने तैयार किया, लोगों ने हमको जानकारी दी, हमने पुलिस पे रिपोर्ट की, उस बात में मामला आगे बढ़ा। तो जब हम किसी घटना की तह में लगातार जाते हैं तब हमको धीरे धीरे सफलता मिलती है। कुछ गांव में ऐसा भी हुआ कि जब शुरू में लोग हमारी बात नहीं मान रहे थे, यहाँ पर एक अभनपुर के पास एक गांव है, वहां पर एक व्यक्ति ने दावा किया है कि उसके घर के अंदर से एक मूर्ति निकली है और उसका एक कागज में लिखा था कि मैं खुद प्रकट हो रही हूँ। जब हमने वहां जांच की, हमने वहां के टीआई का फोन आया, लोग वहां जाने नहीं दे रहे थे। हम लोग जब गए वहां पर तब हमने देखा तो वहां पर उस जो व्यक्ति था उसने हमने गाँव वालों से कहा कि हमको सिर्फ 2 मिनट के लिए देखना है। हमने देखा कि उसने एक जो घर में जो मसाले पीसने वाली जो सील होती है उसमें उसने आंख वाँख बना करके उसने उसका प्रचार किया था। जब हमने गांव वालों को मूर्ति को बाहर निकाल करके समझाया कि भाई ये तो आपको धोखा दे रहा है, तो जो गांव के लोग हमको जाने नहीं देना चाह रहे थे वो लोग हमारे को साथ में हम गए और उसकी पुलिस में रिपोर्ट लिखाई। तो जब हम आगे बढ़ते हैं तो शुरू में थोड़ी सी समस्या होती है। कई बार गांव में ऐसा भी होता है कि मेरे कुछ दोस्त जो हैं जो डॉक्टर हैं उनको लेकर के मैं गया किसी गाँव में। कई बार हमने बहुत सारे सरपंचों को जोड़ा है इस पे, हमने कई पुलिस थानों को जोड़ा है हमने, और हमने यहाँ पर छत्तीसगढ़ में जो कानून बनवाए हैं वो हमने करीब 10 साल प्रयास किया था कि इसमें यहाँ पे कानून बने, और उसके अंतर्गत भी बहुत सारे एक्शन होते हैं। लोग आकर के हमारा व्हाट्सएप नंबर रहता है, तो शुरू में तो लोग बातों को मानते नहीं थे। गाँव में जाने में भी ऐसा लगता था कोई जाएगा नहीं जाएगा, कई बार अकेले भी जाना पड़ता था, रात में गाँव अपनी कार में चले गए, अपनी गाड़ी में चले गए। लेकिन धीरे धीरे अब हमारे पास में एक नेटवर्क है, संगठित लोग हैं, हमको मीडिया का सपोर्ट मिलता है, ऐसी बातों पर प्रेस का सपोर्ट मिलता है, लोग हमारी बातों को समझते हैं और हमारी प्रतिक्रिया लेते हैं, तो ये लगातार चलते रहता है। लेकिन अब धीरे धीरे इसमें एक पहचान बन गई, इसलिए हम उतनी उसी पर संकट नहीं आता है। शुरू में कई बार एक घटना ऐसी हुई, लोगों ने कोशिश किया था कि हम पर हमला भी कर रहे हैं, उन्होंने कुर्सी उर्सी उठा ली थी, लेकिन बाद में उनको लगा कि नहीं भाई ये लोग सही कह रहे हैं, तो वो बात हुई। लेकिन आज तक उसके बाद में कभी ऐसी 1996 में हुआ था ये, उसके बाद कभी ऐसी घटना नहीं हुई, और अब तो ऐसा कोई हिम्मत भी नहीं करता है इस प्रकार से कुछ विरोध करने की।
Thank you, sir.
Thank you, Sunita. Thank you, sir, very much. आज इतने अच्छे टॉक के लिए, जो कि समाज की कुरीतियों के अगेंस्ट काम कर रहे हैं आप लगातार। आपने अपने टॉक में बहुत अच्छे से अंधविश्वास को समझाया, जिसे आपने कहा कि बिना जाने बुझे किसी बात को मान लेना अंधविश्वास है। आपके बहुत ही अच्छी तरीके से यह बताया कि समाज में पाए जाने वाले जो सामान्य अंधविश्वास हैं, जो कि हर तबके के लोग, चाहे वो पढ़े लिखे हों चाहे अनपढ़ हों, मानते हैं, जैसे कि छींकना, चमत्कारिक अंगूठी पहनना, पीलिया का झाड़ फूक कराना, टोनी बिल्ली का रास्ता काटना इत्यादि, मिर्गी आना, बलि देना, ये सारी चीजों पर आपने ये बताया कि ये जो अंधविश्वास है इनके कई कारण हो सकते हैं, लेकिन सबसे सही कारण जो है वो है सही मार्गदर्शन का अभाव। यह एक सबसे बड़ी बात है जो कि अंधविश्वास इतना पनप रहा है। आपने ये भी बताया कि आज जो मीडिया हमारे सामने है वो भी बहुत प्रचार करता है अंधविश्वास के लिए, टीवी सीरियल्स जो हैं, और कई तरह के, जैसे मंदिर की बातें आपने कही कि उस तरीके से भी लोग बाग जो हैं अंधविश्वास को फैलाते हैं। तो आपने इससे ये कहा कि वैज्ञानिक दृष्टिकोण के कारण की कमी के कारण समाज की में जितनी कुरीतियां हैं या जितने इस तरह के अंधविश्वास हैं वो अगर कम करना है तो शिक्षा का प्रचार और प्रसार करना बहुत ही जरूरी है। और ये केवल आपका तो प्रचार बहुत बड़ा है, केवल आप नहीं, समाज के हर तबके के लोग इस प्रचार प्रसार में लगे, इसकी बहुत जरूरत होगी। आप लगातार इन अंधविश्वासों के अगेंस्ट समाज में कार्य कर रहे हैं और इनका निराकरण वैज्ञानिक पद्धति से भी कर रहे हैं। अंत में मैं यही कहना चाहूंगी कि जैसा दीपाली ने कहा कि दिस टॉक इज आई ओपनर टॉक, तो आप केवल नेत्र चिकित्सक ही नहीं है, लेकिन समाज की आंख खोलने का भी काम लगातार कर रहे हैं। इसके लिए आपको बहुत बहुत धन्यवाद। वंस अगेन थैंक यू वेरी मच सर। थैंक यू।
Thank you, sir. Thank you, ma'am.
थैंक यू।
So the fourth and the last technical session is a family matter for us, because we have a learned faculty member of our college, Dr. Sunita Batra ma'am, as our guest speaker. I request Dr. Sangeeta Vajpayee ma'am for formal introduction of our speaker.
थैंक यू दीपाली। सबसे पहले मैं IQAC और रिसर्च कमेटी को ये बधाई देती हूँ कि उन्होंने इतने अच्छे टॉपिक पर जो है ये नेशनल वेबिनार का आयोजन किया है। और मुझे मेरे कलीग, मेरी छोटी बहन का इंट्रोडक्शन देने के लिए जो है मुझे ये मौका दिया है, इसके लिए मैं बहुत बहुत धन्यवाद देती हूँ। It's my privilege to introduce our distinguished speaker of session, Dr. Srimati Sunita Patra. Dr. Sunita Patra is an assistant professor botany at Government NPG College of Science, Raipur, Chhattisgarh. With more than 33 years of teaching and research experience, she is MSc, MPhil, and PhD. She also has diploma in sericulture, diploma in French in her credit. She has published more than 47 research papers and 6 book chapters in reputed publications. She is a well-known research supervisor and has guided 10 PhD scholars successfully. 4 research scholars are currently pursuing their PhD under her guidance. She has qualified NET. She is Fellow of Indian Botanical Society and life member of several renowned organizations. She is an approved counselor of MSc program at IGNOU. She is an accomplished speaker and has delivered more than 27 expert talks. She has successfully organized 11 workshops and has participated in 35 workshops, 15 faculty development programs, and 120 conferences. Her field of interest are biotechnology, molecular biology, microbiology, cytology, and genetics. आज का इनका टॉपिक है सिकल सेल अवेयरनेस अ साइंटिफिक अप्रोच। मैं मैडम पात्रा को बुलाती हूँ इसके लिए टॉपिक।
थैंक यू मैम संगीता वाजपेयी इतने अच्छे इंट्रोडक्शन के लिए। इंट्रोडक्शन ऐसा लग रहा था किसी और का दे रहे हैं, अब मेरा नहीं। इतने अच्छे तरीके से दिया है आपने इंट्रोडक्शन मेरा। सो आई वुड लाइक टू एक्चुअली शेयर माय स्लाइड्स बिफोर स्टार्टिंग। स्लाइड्स विजिबल है?
Yes, ma'am. Visible. Visible.
ओके,
स्लाइड शो पे मैडम, स्लाइड शो पे जाइए।
okay. Is it okay?
Yes, ma'am.
So today's topic is sickle cell awareness, a scientific approach. ये टॉपिक हमने क्यों लिखा है? Because the topic of the seminar is scientific social responsibilities. So what are our responsibilities toward our society? If we are a science students, if we are a faculty of science, and there are certain responsibilities. So suppose if you know science behind some of the diseases, some of the causes, some of the superstitions, we should we should go to the root cause of that and should spread how can we get rid of it. So today's I have chosen this type of topic, sickle cell awareness. Before most of the students, they are also the part. So first I will talk about what are the basics of sickle cell anemia, why the red blood cells, normal red blood cells, they are converted into sickle cell. And then what are the science behind it, how it can stop spreading this type of diseases, what are the myths behind it, and some of the approaches what we are doing in our institutional level. So this was the first case. I think the slide is moving. This is the first case when a dental student was identified in 1910 with a sickle cell diseases. Okay, and now you can see the map of India. The entire the dotted spot where the red dot you can see here, where the sickle cell is found, whether the patient of sickle cell has been identified. So it is entire Chhattisgarh. So we are from Chhattisgarh, therefore we are basically interested in Chhattisgarh, and entire Chhattisgarh has been found be suffering from this disease. Then Western Odisha, Eastern Madhya Pradesh, East Maharashtra, Eastern Gujarat, North Telangana, Kerala, Tamil Nadu, Karnataka, few regions. So we can say there's this sickle cell diseases. It is concentrated more on Chhattisgarh, but we can find in all over India, we can see sickle cell patient, that patient suffering from sickle cell. Now, what is sickle cell? So as we know, we are students of science, and this webinar, the Science College is organizing this webinar, and we teach our students what is DNA, what is chromosome. So, sickle cell disease is an autosomal recessive disorder. So you can see hemoglobin gene which is present here. So abnormal there is a mutation in the hemoglobin gene, and due to this mutation, the normal sickle cell, the normal shape of the sickle cell, it undergoes accretion shape, which is known as sickle. Now, to go into more depth, how this, what is the mutation? What is the cause behind this mutation? So if we see the normal mRNA, you can see the codes. There are triplets. Students, they can well recognize that mRNA, the language. Even Dr. Rath has also started from right from the central dogma, the message they are present in the DNA and they are conveyed to RNA, and RNA then to polypeptide chain. So this is the central dogma. So this is what I'm showing, the sequence of mRNA, messenger RNA, and triplet of nucleotides, we call it as codon. So highlighting here is green color, that means GAG. It's a amino, it's a code for amino acid, glutamine. Now, if there is a point mutation, what's the meaning of point mutation? That means is any one of the nucleotide, it's get mutated. Yes, you can see the seriousness. There are thousands and millions of nucleotides which are arranged, and they are actually the languages or of a phenotypes of metabolic disorder, whatever it is. So if any one of the nucleotide, it gets altered by mutation, in the case of sickle cell, we can see mutation, it occurs, and adenine is converted into a uracil. So AGA with codes for glutamine is now GUG with codes for valine. It is the 6th position you are seeing, glutamine. It is a negatively charged amino acid and is replaced by valine, which has no charge. So this is the molecular basis. What is the mutation? What we say is mutation and sickle cell disease behind it. Now, you can see a normal HbA means it is a normal one. So the chain of, it is the 5th amino acid, that is glutamine, which is converted into valine. And due to this conversion, you can see here that normal red blood cells, they are converted into sickle cell. A single amino acid where glutamic acid is replaced by valine in the 6th position of 146 amino acid of beta chain of hemoglobin. Now, what's the function? Why we are talking about what is sickle cell? Why we are talking about beta chain of hemoglobin? So what is the function of that? Its function of hemoglobin is to carry oxygen. Normally, hemoglobin has 4 units and contains an oxygen binding site. You can see the PubMed structure here. Now, in sickle cell, what we see that there is a mutation. So there are 4 subunits. We can see there are 2 alpha and 2 beta subunits. Hemoglobin is composed of 2 alpha and 2 beta subunits. Then what actually happens when a glutamic acid is converted into a valine? Now you can see polymerization and depolymerization occurs. Polymerization in tissues, in oxygenated sickle hemoglobin. That means if there is ample amount of oxygen, so oxygenated sickles, they are round, normal. Now, deoxygenated hemoglobin, what it actually happens, the hemoglobin, they get polymerized. Yes, you can see the polymerization here. In deoxygenated condition, there is polymerization. Polymerization of deoxygenated sickle hemoglobin, you can see in various tissue. And again, when it is oxygenated, this depolymerization is converted. If it is reoxygenated, we can see here. So polymerization and depolymerization. When there is ample of oxygen, then there is depolymerization. When oxygen is deficient, there is a polymerization and again depolymerization. This is the basis. Now, moving further, we can see that if there is if there is 0% polymerization, what actually happens, that is polymerization of hemoglobin cells. You can see here polymerization. As the oxygen saturation goes up from 40% to 70% to 100% depolymerization, and vice versa. So it depends upon saturation, oxygen saturation. Now, what is the mechanism of sickle cell formation? So SBS means sickled hemoglobin. There is no problem when it is oxygenated. Problem occurs when there is deficiency in oxygen. In oxygen deficient condition, sickle hemoglobin, it polymerizes to form taccoid or fluid polymers, and this polymer realign, and thus RBC is deformed. And this deformed RBC, they form a sickle shape. So sickle shape, the sickling depends upon what? Decrease in oxygen level, pH, and dehydration of patient. So this you can see, it is the normal hemoglobin, but in deoxygenated condition, what actually happens? There is polymerization and there's formation of inflexible chain-like structure. So this chain-like structure, this polymerization converts normal oval sickle RBCs into sickle RBCs. Normal RBCs are compact, flexible, unable to squeeze through small capillaries, which is very important. But what actually happens in sickle RBCs, they are stripped, angular, causing them to become stuck in small capillaries. So deficiency of oxygen. So yes, we can here see sickle cell polymerization occurs. This is the oxygenated condition where we can see. And then in deoxygenated condition, what actually happens? There is polymerization, and this polymerization, the sickle cell, it develops a stickiness, a stiffness, blockage of capillaries. Then occurs vasooccultation, reduced or non-availability of oxygen, then inflammation. These are the clinical symptoms which are the consequences of conversion of normal RBCs to sickle cell RBCs. So normal RBCs, it contains GAG, normal glutamic acid, sickle RBCs. So here we can see the polymerization and depolymerization which occurs, and this sickle RBCs, they are more aggregated, sticky, they stick to the capillary wall. Then there is loss of blood flow and the consequences. So this is the summary what actually happens. Sickle gene, GAG, is converted to TG, result in deficiency of hemoglobin. That when exposed to deoxygenated condition, it polymerizes, resulting in the formation of sickle cell. And what actually happens? Vasoocculation then occurs. So the disorders are also characterized by abnormal adhesive property of sickle cell in peripheral blood mononucleosides. So this aggregation, these mononucleosides, they have receptors, and I'm not going to more in the molecular part, because we are talking about how we can spread this scientific talk, how we can spread this knowledge to the students. But for this, they should know what actually are the root cause of this sickle cell. So sickle erythrocytes, they can also adhere directly to the endothelium cells, abnormal movement or rolling and slowing of cells in the blood tissues, it occurs. This resulted in endothelial damage. The sickle cell also become dehydrated as a result of abnormal channel. And then there is hemolysis. So because the normal cell RBCs, their shelf life is more long, and their sickle RBCs, they are short-lived. So hemolysis contributes to oxidative stress, dysregulation of arginine metabolism, both of which leads to decrease in nitric oxide, that in turn contributes to vasculopathy and then characterizes sickle cell diseases. So this is normal cell, and the life for it is 120 days in normal condition. But in sickle cell disease, the red blood cells only last an average of 10 to 12 days. So the origin of disease is a small change in protein globulin. The change cells arises from a damaged structure of hemoglobin, And a single chain in amino acid, it can cause hemoglobin to aggregate, to form polymerized, and thus leading to sickle cell, which aggregates the vessels. Now, pathophysiology of sickle cell disease, genetic defects, changes the shape, premature hemolysis, and increases the viscosity, adherence to endothelium, microvascular obstruction, and then infraction, vessel occlusion, and organ damage. Sickle cell removed by spleen, they are damaged, and then infection. So these are the consequences what actually happens. So when we compare normal RBCs with the sickle cell shaped, so starting with the disc shape to sickle shape, soft, normal RBCs, they are soft, sickle RBCs, they are hard, like a piece of wood. Normal acids, they can easily flow through small vessels, but sickle cell, they often get stuck in small vessels. Normal RBCs lives for 120 days and sickle RBCs, they live for 20 days or less. So we have talked about how there is a free flowing of normal RBC, and then the sickle RBC, they get stuck in the blood vessels, lack of RBCs and oxygen deficiency in the cells, in the tissue damage. You can see here, yeah, how these normal RBCs, they are free flowing, and the sickle cell, they just get constructed, adhere to the vessels, and the free flowing of the blood, it stops. And if there is no oxygen, hemoglobin is the oxygen carrier. So if there is no oxygen, then pain occurs and organ damage occurs. Consequences, we can see the consequences here. And due to this, you can see the symptoms of sickle disease. This sickle cell disease, it can affect every part of the body: bone pain, abdominal pain, fever, acute chest syndrome, perispasm, deep jaundice, leg ulceration, nervous system. So it affects each and every part of your body. So this is a slide just to see how the sickle cell, it actually damages, how it is circulated, how it is circulated throughout the body, and what actually happens. No, retinopathy, pulmonary hypertension, acute chest syndrome. So again, chest, septicemia, all these skin ulcers, chronic pain, necrosis, delayed puberty. So these sickle hematopathy, meningitis, stroke, neuroconjunctive dysfunction. So each and every part of the body, it is affected by this. Now you can see some of the patient's symptoms: typical face, low hemoglobin, breathlessness, pallor, irritable, exhaustion. You can see these are the symptoms of anemia. Yes, you can see hand and foot syndrome, painful crisis, massive enlargement of spleen, pruning of blood, spleen, shock-like condition. Now, this slides I have taken from Department of Biochemistry, Dr. P.K. Patra. He has provided me these slides because they are studying the patient with sickle cell disease, chronic leg ulcers. You can see here. So it is a very alarming situation with sickle cell patients. The ulcers, they won't heal up very certainly. Then the brain stroke due to sickle cell diseases. They are studying with this type of. Now, managing sickle cell, this we can do up to, so this is in our hand. How can we manage sickle cell? What we can do, what we cannot. So fluids, antibiotic, rest, pain medication, prevention of infection, healthy lifestyle, and counseling. So what is our part? What educational institution can do to prevent sickle cell from spreading? So it is the counseling part. How we can go, how we can counsel our students scientifically, how we can convince our students for this, so they can spread this knowledge to their home, to their parents, to their relatives, to their villages. And this is our role for counseling. So there are certain treatments, cure of sickle cell disease. So there are treatments: bone marrow transformation, prevention by hydroxyurea. The doctors, the physicians, they are taking care of it. So treatment of sickle cell anemia, doctors, they take care of it, the folic acid preparation, hemotransfusion, analgesics, erythropoietin, immunization, hydroxyurea, and antibiotic intravenous rehydration. These are the symptoms. The treatments of sickle cell disease, then there are gene therapy for sickle cell. The gene therapy is still going on. The collection of blood and transplantation of corrected stem cells into the patient. This is also a therapy, gene therapy for prevention or curation of sickle cell, we can see. So this is how it happens, direct delivery and cell-based delivery. So we can put the healthy RBCs, the stem cells to the cell, transplantation of hematopoietic stem cells to the patient, and then we can go through it. But this treatment is very expensive. So yes, we can go. Then there is the latest technique, which is known as a gene editing technique, CRISPR-Cas19. What is CRISPR? Clustered regularly interspaced palindromic repeats. And this is actually used to edit a sickle cell mutation. What we have seen, that point mutation where the single nucleotide is get altered. So this can, now the researchers is going on CRISPR-Cas for gene editing of sickle cell. Now, what is in our part? In our part, the educational institution, they can go for counseling. Now, आप ये देख सकते हैं जिसमें लिखा हुआ है अभिभावकों, शिक्षकों, सहपाठियों, और इनका क्या रोल हो सकता है, हम क्या कर सकते हैं सिकल सेल पेशेंट, क्योंकि ये तो हेरिडिटरी है. ये जो सिकल सेल अगर किसी को होता है तो इट इज इनहेरिटेड इन नेक्स्ट जनरेशन. और सो हाउ वी कैन स्टॉप फ्रॉम इनहेरिटिंग दिस डिजीज? हम क्या कर सकते हैं? हम उनको, वी कैन गिव देम साइंटिफिक एडवाइसेस. एंड इफ ए पेशेंट इट इज सफरिंग फ्रॉम सिकल सेल, व्हाट वी कैन डू, वी कैन गिव द पेशेंट्स, हम स्टूडेंट्स को हम क्या दे सकते हैं? पुस्तकें बांट सकते हैं which deals with the knowledge of sickle cell. Then हमेशा जो स्टूडेंट्स जो है वो, अगर उनको सिकल सेल पेशेंट्स हैं, हमेशा क्या करना है? उनको पानी की बोतल रखना है, क्योंकि डिहाइड्रेशन होता है इससे. तो बार बार आपको पानी पीते रहना चाहिए. नो, व्हाट एक्चुअली हैपेंस ड्यूरिंग दिस, अगर विद्यार्थी क्लास में है, बार बार पानी पी रहा है, तो मे बी ही कैन आस्क फॉर यूरिनेशन. और कई बार टीचर्स जो है इससे इरिटेट भी होते हैं, तो वी हैव टू बी पेशेंट. अगर कुछ बच्चे बदमाशी भी करते हैं, but we have to ask if you are suffering from this disease or you have any problem, any complications. So we should allow them to drink water and to go for urinal. Then बहुत ज्यादा काम नहीं करना है, because we have seen that the sickle cell जो है वो deoxygenated condition में होता है. तो आप ज्यादा काम करेंगे तो इससे आपको दिक्कत हो सकती है शरीर में. तो इसलिए ऐसे स्टूडेंट्स को, ऐसे विद्यार्थियों को उसके लिए प्रेशराइज ना किया जाए, उनसे ज्यादा परिश्रम ना हो सके. दूसरा, बाहरी वातावरण से सुरक्षा. तो ऐसे सिकल सेल पेशेंट जो है गर्मी में, बहुत ठंड की स्थिति में बाहर नहीं जाना चाहिए, क्योंकि इनसे तापमान जो है खून के बहाव में परिवर्तन ला सकता है. तो इन सब के बारे में जानकारी हमको देनी चाहिए, हमको ये स्प्रेड करना चाहिए, ये टेम्पलेट्स वगैरह वितरण करना चाहिए. और इस तरह से हम शिक्षकों को, द टीचर्स, द फैकल्टीज और जो फ्रेंड्स हैं, उनको इसमें मदद करना चाहिए. मान लीजिए अगर सिकल सेल पेशेंट किसी क्राइसिस में आता है, तो उसको डायरेक्टली हमको हॉस्पिटल लेके जाना है, ये हमको ध्यान में रखना है. तो ये बहुत इंपॉर्टेंट है. और ये पैम्पलेट जो भी है, ये सिकल सेल प्रोजेक्ट द्वारा बायोकेमिस्ट्री विभाग, पंडित JNM मेडिकल कॉलेज द्वारा हम लोगों ने इसको लिया है. और जब भी हम इसको काउंसलिंग करते हैं तो ये हम वितरण करते हैं. वी डिस्ट्रीब्यूट दिस पैम्पलेट टू द स्टूडेंट्स सो दैट दे कैन go through it. Now, a very part, how we can spread, हमारा क्या रोल है, हम किस तरह की काउंसलिंग कर सकते हैं स्टूडेंट्स को. सो नॉर्मल, यहाँ पर जो स्लाइड्स में दिख रहे हैं, we can see AA is normal. AA is normal. We have one set of gene. So there are one is normal and one is sickle. So this is known है. इस तरह का अगर आपका तो ये इस तरह के अगर आपका है, तो वी कैन से दैट दे आर कैरियर, वाहक है. ये सिकल सेल नहीं है, बट ये आगे चल के सिकल सेल पेशेंट्स को जन्म दे सकते हैं. सो दे आर कैरियर ऑफ सिकल सेल, बट दैट मीन्स वन ऑफ द जीन इज म्यूटेटेड एंड वन ऑफ द जीन इज नॉर्मल. नाउ रोगी, दैट मीन्स द सफरर, व्हाट वी से, दैट मीन्स बोथ ऑफ द जीन्स दे आर म्यूटेड. सो इसको हम कहेंगे दे आर सिकल सेल डिजीज. सो सिकल सेल डिजीज जो पेशेंट्स होते हैं, जो कैरियर्स होते हैं, सो उनको बहुत ज्यादा ख्याल रखने की जरूरत है। जो एस होते हैं, बिकॉज दे हैव वन नॉर्मल जीन एंड वन जीन इज म्यूटेटेड, so they can live healthy normal life. There is nothing to worry about. Now, how these genes they are transmitted? You know the Mendelian cause. You can see here the normal gene and the sickle cell gene. So if there is a carrier, you can see red one is the mutant and blue one is the normal. So if both the parents are having a trait, that means both of the parents, if they are carrier, you can see the children, we can see normal children with no sickle cell. We can see there are two children with sickle cell trait, that means they are the carrier. And again, there is very possibility to have a child with disease, but that means both of the genes, they are mutated. Genes, they can be transmitted. So this is the genetic heredity, what we study in our classes. The students, they should get acquainted with what type of heredity, how the genes, they are transmitted from parents to children. So counseling part, what we do in our counseling part, suppose we actually show this diagram to our students. That means first thing is, before going to counseling, what we have to do, we just go through lecture series of a renowned personality who have already worked on sickle cell. So that they can give the sickle cell, then we go ask them for the screening process. And after screening process, we go for counseling. If the suppose a student has been detected with suffering from sickle cell, so we go for counseling. So the cards, they are distributed there. So supposing that they are AS or S, that means whether they are carrier, whether they are sickle cell or they are normal. So we counsel them that if both the parents don't go for marriages if both are carriers, because we have very possibility that the child may be carrying maybe disease, both of the genes, and they may have, the child may have both of the mutant genes. So this is sickle cell. So usual trait, suppose if there is marriage between a normal and a carrier, so we can see that we have normal, then two of the children, they are normal and two are carriers. That means four of them, they can lead a very healthy and normal life. So this type of counseling we give to the students. And this is also, we call it as sickle cell kundali. So before marriage, this type of pamphlet are also circulated to our students, because they know what, so what they should do, what they should not. So so red one shows the sickle cell and the green, it shows the normal gene. So whether the parents both are normal, there is nothing to worry about. One of the parent, it is normal and one is carrier, again it is nothing to worry about, because if the child, they are it will be always a carrier one. So it will live he or she will live a very normal life. But alarming situation is when one of the parent is a carrier and another one is sickle cell disease. Don't go for the marriage, because you have very possibility of having your child with sickle disease. And likewise, we distribute this template to our students. So scientifically, the students know why providing, why we are inhibiting, why we are discouraging such type of marriages. Now, there are some couple of slides what we are doing in our institute to do this. So first of the slide is slides of Dr. P.K. Pathak. We should, because we are not doctors, we are not physicians. So whatever we do, we consult our doctor, the physician, because he's working with sickle cell patient last 30 to 35 years. So we frequently call in our college for his lecture and also for counsel purposes. So these are some of the glimpses of this educating students, educating the basic need, how the disease is understanding the genetics about it, how it can be stopped by going to next generation. And after going to, we organize camp, screening camp and counseling camp for the students. So these camps are not only for students, they are for faculty members, for our office staff, everyone out of college, society people, they can also join. So there is a registration process, and you can see there is a well-equipped team, which we, Dr. Patra's team, the sickle cell team, to join us, to extend their help, and with their help we have organized screenings and camp and counseling process. At first it was really, the students were not actually able to understand why we should go for sickle cell screening. They were hesitating. No one of the students, they were coming to give their blood, because it needs only a drop of blood to take, and then there is solubility test and a card is issued. Within a few seconds you can see whether they are sickle cell, a carrier one. So after going through whether they are normal or sickle cell, the sample, they are sent to the Department of Biochemistry for electrophoresis to see whether they are trait or carrier. That means AS or SS. And they send back us the cards informing that your student is suffering from sickle cell or he is a carrier one. We distribute this card among to the students. And if the student is actually a sickle cell disease, we give them pamphlets, we give them then marriage counseling. Now the students, they are very much aware of that. So they come to me, they ask about if they are suffering from, how we can get medicines. So medicines are distributed free of cost in government. So these are the symptoms we can see here. We can see this queue of students, they are very enthusiastic. And when we explain the student a scientific way, it helps this scientific information to the society. These students, they go to their home, they explain this disease to their parent, and so on. From our part, we are doing this to help prevention of this disease. As you can see, and it's not limited to our college campus. We go to villages where we organize a camp with the help of Sickle Cell Project with medical professionals. We took them there. You can see here the line of students, their card, and all their medical profession, then height, weight. And you can see in one of the slides that students, first we actually taught our students, and now students, they interact with the students of the school children. So such on the information is passes from students to students. This is another camp which we have organized in another village. You can see here. So it was very surprising. We sat there for right from 10:30 a.m. and there were no patients, no students to come over. And then as soon as the school students, when they come, they got the information, they went back to their home. And then now you can see they brought their parents, their grandparents for screening purpose. So it was really very enthusiastic one. And yes, this is very one, you can see here Dr. Lalji Singh, one of the renowned scientists. I went through, I was attached with him till 2008, 2009. So he's also pioneer of DNA fingerprinting, popularizing DNA fingerprinting in India. So going to the college, go at the basic level, then interact with the patients, interact with the tribals, with the villagers, and we come back at the night and doing the scientific work here. So it was a very good exposure. And again, there was again the camp. So again, the courtesy is of Government Medical College. We join their team, we go there, we spread information. So this is the counseling, that medical team. They, even if there is need of any medical expert, in the team, they have medical professionals, the physicians, they give them a proper advice. So together we can do it. So this is the solubility test which is done. Then there are certain apps also. So with the help of apps, patients can also can interact. So all this helps there. And we actually, we are fortunate enough because we have MOU with this institution. So they are really generous to help us in all this. So this is all about sickle cell disease, the awareness, and how we can spread the awareness scientifically among the students. Thank you.
If you have questions, you may ask.
Any question?
Ma'am, I have one question, ma'am. What is the current status of gene therapy in case of sickle cell disease in our country?
Yes, gene therapy is going on. CRISPR-Cas is the latest technique. Now, Dr. Patra's team, they are also going through it. They are doing.
Okay.
And in very few couple of days, we can see the results. Yes, there are many ethical issues also. So we have to take care of it.
Okay.
So works are still going on.
Thank you so much, ma'am, for sharing your knowledge. Sickle cell is not unknown to our state as it is one of the diseases debilitating lives and working capacities of people. With proper awareness about the genetic based disease, its spread can be minimized. And also early detection and therapeutic approaches can be applied for the betterment. Thank you, ma'am. I invite Dr. Sangeeta Bajpayee, ma'am, for vote of thanks.
थैंक यू डॉक्टर पात्रा। सबसे पहले तो मैं कहूंगी कि आपने बहुत ही अच्छे से और बहुत ही सिंपल वे में पूरी कैसे सिकल सेल क्या है और हमारे छत्तीसगढ़ में सिकल सेल कितना सीवियर है और सिकल सेल कैसे होता है, सिकल सेल के क्या क्या पैथोफिजियोलॉजी है, क्या उसके सिम्टम्स हैं, कि किस तरह से आपको हाइपरटेंशन होता है, स्किन अल्सर हो सकता है, पेन हो सकता है, स्प्लीन एनलार्जमेंट है, ये सारी जो सिम्टम्स हैं उसको हम किस तरह से मैनेज कर सकते हैं एंटीबायोटिक, रेस्ट या हेल्दी लाइफ स्टाइल से। और सबसे बड़ी चीज जो है काउंसलिंग, उस पर आपने सबसे ज्यादा जोर दिया है कि और काउंसलिंग से जो है बहुत सारी चीजें पहले ही हम जो है क्योर कर सकते हैं। और ये हमने मैडम की सहायता से और डॉक्टर पात्रा की सहायता से कॉलेज में कई बार हमने जो है वो सिकल सेल स्क्रीनिंग रखी है और उसका हमने बहुत ज्यादा बेनिफिट भी देखा है कि पहले शुरुआत में जब हमने पहला रखा था तब बच्चे स्टूडेंट्स बहुत अवेयर नहीं थे, वो ब्लड टेस्ट कराने से ही डर रहे थे। पर सेकंड टाइम जो हमने रखी थी स्क्रीनिंग, उसमें बच्चे खुद आए, मैडम से उन्होंने कांटेक्ट किया उसका, और बकायदा से मेडिकल कॉलेज में जाके उन्होंने ट्रीटमेंट लेने की भी मतलब पूरी आगे वो प्रोसेस करते रहे। तो जो मैडम ने कहा कि काउंसलिंग सबसे ज्यादा इम्पोर्टेंट है इसमें, क्योंकि हर स्कूल और हर कॉलेज में, हर जगह, स्कूल कॉलेज के अलावा सोशल लाइफ में भी हर आदमी के पास जो है वो सिकल कुंडली की जानकारी होनी चाहिए, जिससे कि ये डिसीज जो है वो आगे की तरफ ना फैले। तो मैडम ने बहुत ही अच्छे से जो है इसकी जानकारी दी। तो थैंक यू डॉक्टर सुनीता पात्रा।
Thank you, ma'am, for your kind words. I can see Luis Benaberto, our speaker of tomorrow's speaker. Yes, sir is there. Welcome, sir, in this our session. This was the concluding part. सर जुड़े हुए हैं, अभी मुझे दिख रहे थे।
I think he's gone somewhere. उठ के गए अभी।
हाँ सर, okay.
हाँ।
Okay, you can go forward, I think.
So we have reached the conclusion of this webinar's first day, ma'am. So shall we conclude or should wait for sir?
I think sir were there and I think कहीं उठ के चले गए हैं, and we should conclude the session.
Fine. Okay. So with this, reach the end of day one of this webinar. I hope all the participants had good time listening to our eminent speakers. We should all connect tomorrow at 12 sharp for the next day. And then good evening, everyone.
Okay, Deepali, just give highlights of tomorrow's session. If you have the schedule, you can go
Yes,
to
ma'am,
it.
yes,
Or
ma'am. Just a second. Just a second, ma'am. So tomorrow we'll be beginning at 12 o'clock
Okay. So there is connection problem with Deepali. So in tomorrow's session, that is 20th of January 2024, there is our technical session 3. It will start from 12 sharp. So our first speaker for tomorrow will be Sadhana Sharma. And his talk is how science impacts our daily activities. Then we have Dr. Varnika Sharma.
Yes, ma'am.
Okay.
Then we have for the valedictory session, we have Dr. Anil Kumar from JP University, Chhapra, Bihar. We'll be talking about
Hey, there is
social
Varnika.
implications of chronobiology. This is about the technical sessions we'll be having tomorrow. Was I not audible, ma'am?
Yeah, you are audible. And after that, there will be summary of seminar, valedictory speech by our principal. I think sir is there. Yes. Welcome, sir. Barato, sir. Welcome in our webinar. I think we should conclude the section.
Yes, ma'am. Yes, ma'am.
Okay.
It's morning there. Maybe he's not getting that we have reached the conclusion of our seminar.
Yeah.
अपना वीडियो ऑन कर लेते तो फोटो एक अच्छी हो जाती.
हाँ, all the participants who are there, please put your video on. Yeah, again. Welcome, sir. Okay. So with this, we come to the end of today's webinar. And tomorrow we will gather here again to start a new session at 12 o'clock. Thank you very much, everyone, for joining us today. Thank you. Now you can leave this session.